pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Oh, it's not on. Sorry. Somebody gave me a thumbs up that my mic worked. They lied to me. Okay, now you can hear me online. Thank you. Okay, so I'm not going to reiterate all of that except for the fact that I'm Pete Weina, and welcome to the second day. Hopefully we won't have as many of the glitches that we had with Adobe yesterday. I've been assured that all of it is fixed and everything's going to go very smoothly today. So logistics, please make sure that you evaluate each session. Those of you that are in Adobe Connect will have a link in front of you that you can click on. Those of you joining us via YouTube, unfortunately, you won't have an active link that you can click on, but welcome to our informational session anyway. Overall, evaluations, make sure that you do the overall evaluation for the entire session. And if you have not signed in, if you were here yesterday and you came back again today and just bypassed our front desk, make sure that you stop at the front desk sometime during the day today to sign in so that you get credit for this and that you can get a certificate of attendance if that's important to you. The link will be activated and sent in the post-evaluation event thank you email for all of those that are registered. And upon completion of the survey, an attendee can then download a certificate of attendance and we'll get you credit in a variety of different organizations. That certificate of attendance, I've been told, is only available at the time that you do the evaluation. So if you don't download the certificate of at that point, it will kind of vanish into the background. So make sure that you download that certificate at the same time. And it will only be available until June 3rd. After that, that link will disappear. And recordings will be posted to the event website once available. I looked at, for example, yesterday evening, I looked at some of the other sessions from like CBER and CDRH, and those are already on YouTube for viewing. So With that, I'm just going to turn it over to my colleague, Sharu Mullick from Dr. Sharu Mullick. She is our acting. She made sure that I brought up the term acting, Associate Director for Labeling and Regulatory Policy, and she will be introducing our two speakers for the first session. Sharu.
Thank you, Pete. Yes, I am Charu Malik. I'm somebody who works in the Office of Vaccines with Pete and with some of the presenters who you'll listen to today. So welcome to session 7. This session is about regulatory considerations for bacteriophage products targeting bacterial infections. Dr. Susan Lehman is a product reviewer and researcher in the Office of Vaccines. She has extensive background experience in phage and bacterial biology. Dr. Lehman reviews CMC, chemistry manufacturing control packages for IND applications for phage treatments and other products in our office and divisions. Dr. Lehman conducts original research on phage activity among other related focus areas. And with that brief introduction, I'll turn over to Dr. Susan Lehman. Come on, Susan.
Thank you. Just make sure. There we go. Good morning, everybody. My title kind of hides the term that most people use when talking about this area. So this talks about bacteriophage products targeting bacterial infections, but the commonly used term is phage therapy. So if you've heard of phage therapy, that's what I'm here to talk to you about today. I am speaking from my experience as a product reviewer, but fundamentally, I'm talking to you today from my own best judgment, and none of my comments bind or obligate the FDA in any way. So what I'm going to take you through today is, that's probably a lot better for the online folks. What is phage therapy? What is, I'm sorry, I don't have, am I supposed to have the screen in front of me? Am I supposed to be able to, because I can't see my own slides without turning around.
Yeah, unfortunately. Okay, so once it turns yellow, it should be coming up.
There, it's starting to come up. Much better. Now I can talk into the microphone instead of at the screen. So I want to take you through what phage therapy is, what the FDA's role in regulating it is, and then talk a little about the kinds of phage therapy applications that we review and spend the meat of my talk sharing how we think about phage products. So first, what is phage therapy? Bacteriophages, phages, these are viral predators of bacteria. And the way that they kill is by attaching to a susceptible bacterial cell, transferring their genome inside the cell, and then taking over the bacterial metabolic machinery to make copies of themselves. At the end of that process, they lyse the bacterial cell and release dozens or hundreds of progeny phages that can go on to repeat this cycle. Phages only infect bacteria. Each phage is usually specific to one or a few species of bacteria, and sometimes even specific to only a few strains. They're found everywhere, including inside of us as part of the human microbiome, and they aren't expected to actively affect human cells. The focus for my talk today and for phage therapy in general is on obligately lytic phages. If you're familiar with phages in other contexts, you may be familiar with temperate or lysogenic phages that can incorporate themselves into the bacterial genome and be passed vertically to daughter cells. That is not the type of phage that's usually proposed for phage therapy. So this talk is focused on obligately lytic phages. Phage therapy is not new. Phages were discovered over 100 years ago and pretty quickly became of interest as potential antibacterial agents. So they were investigated clinically in the 1920s, primarily in Georgia, Russia, and Poland, where work continues to the present day. But there was interest elsewhere, including in the US. That interest mostly waned with the advent of antibiotics in the 1940s and the 1950s. And all of this early work was conducted outside of modern pharmaceutical development frameworks. However, in the last few decades, there's been a lot of renewed interest globally particularly for patients that have infections that do not respond to antibiotics. So infections that involve multi-drug resistant organisms and biofilms. What is the FDA's role in regulating phage therapy? Simply put, phage therapy products are biologics and drugs. Phage therapy as a term typically refers to products that are being given to humans to treat, prevent, or cure a disease or condition. And in that context, we consider them investigational products that are regulated by the FDA under both the FD&C Act, the Food, Drug, and Cosmetics Act, and the Public Health Service Act. At the moment in the US, there are no phage products that are licensed to treat human infections. There are some products that are available for use on foods or in agriculture, but not for humans to treat human infection. There are clinical trials underway. There are patients that are able to access individual treatment, and I'm going to talk in more detail about both of those later on. And currently, product safety is mostly managed via robust phage characterization and product purity. Globally, there are some products that are sold locally for human use in Russia, in the Republic of Georgia, and Poland. These are often for use either orally or topically. Europe, the UK, Canada, and Australia, some other regions also have avenues for legal use of phages in humans that involve clinical trials and some mechanism for individual patient access. What kinds of phage therapy applications do we review? The answer to this stems from the fact that there are two avenues for legal use of biologics in the US. One is that you can have a licensed product. And for phage therapy, this would be a biologics license application or BLA. As I mentioned, we do not currently have any products approved in the US, which means that what we are reviewing now are INDs, or investigational new drug applications. There are two types. One is the regular IND that we're all familiar with, something that supports a series of clinical studies, whether they are research studies or intended for commercial product development. And the other type of IND is called expanded access. So these are intended to treat one or a few patients. It enables them to access treatment with an unlicensed product. That is sometimes called compassionate use, but the formal term in the US is expanded access. And these differ from clinical trial INDs. These are not intended to collect data that would support regulatory action. They're intended to help patients in need. For phage therapy products that have an infectious disease indication, these are typically reviewed within the Center for Biologics and the Office of Vaccines. A typical phage therapy review team will have representatives from our Division of Review Management, from our Clinical Review Division, the Division of Clinical and Toxicology Review, and from a product division. And right now that is the Division of Bacterial, Parasitic, and Allergenic Products, where I belong. We also consult with statisticians, with other CBER offices and FDA centers as appropriate based on the specific aspects of any particular application. Now I want to get into the meat of this and some of the details, how we think about phage products when we are reviewing the product itself. For all of the INDs we review, safety is our first concern. We frame that in terms of the potential benefits and risks, and we assess those benefits and risks based on the context in which the product is going to be used. So that can include factors like the patient population, whether the disease condition that's being treated, whether that patient population is more or less vulnerable to potential side effects. How does the route of administration affect the perhaps sensitivity to impurities, things like that. And then within that context, we then look at the product itself. And there are four main aspects of the product that we are thinking about. The phages themselves, the bacterial production host that they're grown on, because phages are obligate predators of bacteria. If you want to make a bunch of phages, you have to grow them on a bacterial host. And that bacterial host is going to affect the impurity profile of your final product. So that's really important. We look at the production processes that are used and then how you purify that downstream. And we look at the testing. Together, these factors are your CMC, chemistry, manufacturing, and controls. And what it boils down to is we need you to tell us what you know about the product and how you know it, because how you know it affects how confident you are in the conclusions about what you know about that product. So I mentioned there are two types of INDs. I am mostly going to focus on our product expectations for a regular IND, and I'll talk a little bit about the expanded access INDs at the end. So a regular IND feeds into this big picture product development process that we're familiar with, where we have an R&D phase 1, 2, and 3 clinical trials, and then depending on the outcome, this may lead to submission of a BLA, where you're presenting substantial evidence of safety and effectiveness, something that's adequate to support a risk-benefit assessment and licensure. If that BLA is granted, there's the potential for downstream additional information. And the INDs that we review obviously are focused on these phases of development. Across all those phases, again, safety is our key concern. But as you move through the phases of development, there's an increasing emphasis placed on gathering evidence of product effectiveness. There's also an expectation that your manufacturing consistency and assay development will progressively mature across those phases of development, so that by the time you are in the position of deciding to submit a BLA, you have a commercial scale manufacturing process that is robust, that will manufacture the same product every time, and you have fully validated assays that will support that. That whole process and that progressive maturation is what we call phase-appropriate GMP. And to get a little more information about phase-appropriate GMP, I would encourage you to read the guidance that we have on CGMP for phase 1 investigational drugs. The guidance describes an approach that reflects the fact that manufacturing controls that we need to achieve an appropriate product quality, they differ between investigational and commercial scale manufacture, but they also vary across the phases of development. So phase 1 clinical trial manufacturing is not subject to the same CGMP requirements as later phase trials or commercial production. And the guidance can help you see how we think about some of those principles so that you can apply them to your own work. Okay, we've done a lot of setup. Let's talk about some specific phage biology. So I mentioned there are four areas that we look at in an IND. Let's start by talking about the phages. For early stage studies, we expect that phages will be fully sequenced, they will be obligate lytic, non-transducing, and that they are free of recognizable bacterial virulence genes or antibiotic resistance genes. If your product is a cocktail, so maybe your product has 3 phages or 4 phages in it that to collectively provide the spectrum of activity that you want, you should have a reason for including each of the phages in that product. They should each have a relevant activity. And you can demonstrate these properties through various combinations of wet lab data, bioinformatic analysis, things like that. We also see applications that involve both naturally occurring and engineered phages. Genetically engineered phages, we don't think that they necessarily present a different safety concern. The important thing is to tell us what you have engineered, how you did it, make sure that you have given us all this characterization information for the final phage that you engineered, and explain what the property is, why, have some data for why this engineered phage is valuable in your product. We also see INDs for both fixed composition cocktails and what's sometimes called a phage library. So a collection of phages that are all characterized, where the idea might be to choose one or a few phages for each specific patient. Both of those approaches can be investigated under an IND, and we have equivalent expectations for the phage characterization. So the second major area that we look at is the bacterial host, because again, this is what you're going to grow the phage on, and it's going to affect the impurity profile of your product. So again, for early stage studies, we typically expect to know what it is. You know, what's the species, what's the strain, where did you get it from? And we're looking for enough characterization information, usually in the form of sequencing, to know that you'll be able to identify that host accurately during your production process. You want to make sure you're growing your phages on the host you think you are. And to enable us to evaluate the potential safety risks to patients, again, from that, understanding the potential impurity profile. However, there can be nuance in what information is part of any particular package. And that's because what you know can influence, what you know and what you don't know influences how you manage risk. So the example I've given here is about toxin genes. A lot of bacteria, especially, if you're looking, if you want a phage that kills a particular pathogenic bacteria, you're probably growing it on a host that is related to that pathogenic bacteria. You don't necessarily, ideally you don't want to use a pathogenic strain, but the strain may still encode some genes that could present a potential safety risk. So let's say that host encodes a toxin gene. You've sequenced it, you know that it's there. The presence of that in your host genome might be offset by having high confidence that your phage is not transducing, so it's not going to be able to mobilize this gene from your manufacturing environment into your treatment environment. And also by testing to make sure that that expressed toxin is absent in the final product. Alternatively, you might not need to test for the toxin in the final product if you know that the host doesn't encode any toxins. And if you know that if you sequence your host, you find it does encode a toxin, consider deleting it. And then you have addressed that concern in a different way. When we look at the overall phage production and purification methods, some of this goes back to that guidance I mentioned for phase 1 cGMP. At the beginning, we are expecting to see characterized controlled raw materials. I want to highlight that for a product like this, raw materials does not just mean your media and your buffers. Your master stocks of phages and their manufacturing hosts, those are critical biological raw materials. So for this type of product, you don't just have chemical raw materials, you have biological raw materials. We expect to see consistent, well-documented processes, and the level of control and the level of documentation adjusts as you move through the phases of clinical development towards full GMP. We look for materials that present a minimal risk of TSE transmission. That can be addressed by using animal-free media or certified media where any animal origin components come from a certified low-risk region. And we look at the processes that you use to particularly purify to make sure that those are scientifically likely to remove most non-phage substances and that you are developing the data to back up that scientific rationale. There are a lot of different approaches that are capable of removing non-phage substances from these types of products. And each sponsor goes through the process of figuring out which one, optimizing their process and developing the data to back it up. And ultimately, that purification process should put your product in a formulation buffer that is suitable for the final route of administration. I just want to highlight again, at the risk of repeating myself, these requirements evolve substantially in the process of moving from phase 1 to phase 3 clinical development towards that commercial product. And that includes how much supporting data are needed to back it up. And so the last piece I'll talk about for this is the testing. Some of you have probably heard the phrase, you can't test your way into quality. You have to make sure that all of the pieces that come before your final product release testing are are going to engender a quality product. But the testing at the end of that is really important to make sure that your processes have in fact yielded the product that you expect and that you are comfortable delivering. And so that always involves, for biologics, potency, identity, and purity. For phage therapy products, potency is often a simple measure of the concentration of viable particles. At early stage, that can be a total concentration, but as you progress, we expect you to be able to quantify each individual phage within a product if you have more than one. Purity testing has a lot of established test methods, but the ones that are quite relevant for phage therapy products include endotoxin. So particularly if you're growing your phage on a gram-negative host, tend to produce a lot of endotoxin, and that has to be removed. If you're going to give the product IV, usually the maximum allowed amount in the product is 5 EU per kilogram per hour, but that applies to some other routes like inhalation as well. Most of the time, a phage therapy product, we're going to expect it to be sterile, although if you're giving it orally, that, it may be that low bioburden is sufficient. We talked already a little about testing for pathogen-specific toxins, and there are also other residual host cell proteins or residual processing aids that will be important to develop testing for as you move forward. Product stability is important. Got to make sure that the product you're giving to the last patient in the study is as potent as the product that you gave to the first patient in the study. And stability becomes, especially having a lot of leading stability, becomes more important by phase 2. And ultimately for all of these, we look for data showing that how your assay is measuring the thing that you think you does. Data that that your assay is measuring what you think it does. Again, influenced by other information, and all of this will evolve with time. So how does that all come back into this big picture phases of product development? So when you get to submitting your initial IND to run your first clinical study, this is all the kind of data that's going to be in there. It's not the only data that's going to be in that IND, but that's sort of a high level overview of your CMC package. The specific requirements are going to change depending on whether you're submitting a commercial IND or a research IND, and I've put a link up there. Use any of these. If you use these search terms, you should pull up this page that provides some information about those requirements. But generally speaking, you've got a clinical study protocol, information for investigators, and CMC information. Those are key parts of that. Once you submit it, the FDA has 30 days to review it, request additional information if needed, and either allow your study to proceed or place it on hold pending resolution of any deficiencies. That is not a tremendously long review time, and you will have put a lot of work into this. We strongly recommend that you consider having a pre-IND meeting. So this is a valuable opportunity to discuss key aspects of your plans with us before you submit your IND. The guidance document that I've put up there has a lot of good information for how you prepare for that. And these meetings are available to all sponsors, including academic researchers and companies. When you hold one of these meetings, you summarize your CMC and clinical trial plans and then pose questions. Those questions should be as specific as possible and cover key aspects of your IND, and you should provide sufficient background. In the case of CMC in particular, that's what I'm looking at, you should provide sufficient CMC background to allow us to address the specific questions. So, sponsors will get much better, more useful responses by proposing a specific plan and then asking a specific question about that than by asking something open-ended. When you submit the meeting package, we assemble a full review team. We provide preliminary written responses prior to the scheduled meeting and proceed from there. So I mentioned also an expanded access IND, and I wanted to give you a little bit of information about that. The requirements for expanded access are that a patient have a serious or immediately life-threatening disease or condition, and that condition is not being adequately treated with the available therapies. So a patient that is not responding to antibiotic therapy, even though, and all of the available standard of care treatments have been exhausted, for example. The potential benefit should justify the potential risk, and the potential risks should not be unreasonable in the context of that patient's disease or condition. It also needs to be the case that providing the drug won't interfere with clinical investigations that support a marketing approval. So in these cases, the treating physician will submit a form, they submit a summary of the patient's clinical condition, their treatment plan, and the product information. Depending on how time sensitive they consider treatment to be, they can ask for either an emergency expanded access IND or a non-emergency. And in the case of an emergency, sometimes approval can be granted. Sorry, approval is the wrong word. Sometimes we can review and allow that IND to proceed the same day, sometimes the next day. If a case is not an immediate emergency, then it'll be submitted and we have 30 days to review. Although if all of the information is in order in the submission, it often happens faster than that. Our expectations for product characterization in manufacturing are more flexible for expanded access INDs, and that reflects the different benefit risk analysis for patients in serious need. There's some more information at that link. I know for those watching online, the link is long, but again, a keyword search on the FDA webpage will pull that up pretty easily. So my take-home messages are that the FDA is committed to facilitating phage therapy clinical trials to help progress product development. Patients in need can also receive treatment via expanded access. Our evaluation of CMC data is based on science and the context of use for a particular product. And then my last two messages are both about communication. Communication is really important. I do want to highlight that communication occurs between the IND sponsor and in OVRR through the regulatory project manager or RPM. So if there's a team of people working on a particular IND, we can only communicate with the sponsor. So that's make sure you have a designated individual doing that. And then for your product development INDs, definitely recommend talking to us early. I'm happy to take some questions, and thank you for your attention. Yeah.
Please go ahead, yeah.
Mina Izaju, Integrated Pharma. I want to thank you. This presentation has been the best presentation about phage regulations I've ever tried to
Thank you.
find. So we work with several companies and they have different types of phage therapeutics or phage supplements or phage drive products. So I'm going to combine all of my questions into one question. So if a product is not claiming any treatment, does that mean whatever you described does not apply to that phage product?
So that's kind of a tricky question to answer because I would encourage people to think about what the product, what is the product truly aiming to do? If the intent really is to address a specific medical condition, then probably, the regulations apply. If you're not sure what might apply, you can reach out and ask, if the application if it's something that's intended for use in humans, for use in animals, for use on food, you can reach out to the relative centers and ask some questions. So that, if you don't think that something is considered a biologic or a drug, ask questions and be sure. Because it might be. You want to make sure that you understand correctly what applies. And that's not a question that I can answer in this exchange.
Now, continuation of my question. So if someone uses an extract of bacteriophages, endolicense, that is outside of FDA's purview?
Endolicense are a different type of product than what we typically call phage therapy, like whole phages. I believe endolicense are regulated through the Center for Drugs, but you could reach out for more
And last question.
information.
Is there any policy related to use of phages as food supplements, like in Europe?
There are systems that handle use of phage in food. I am not the right person to tell you what those requirements are, but you could reach out to the sections of the agency that deal with food.
Clarification. Food supplements, I mean people taking it as an oral supplement to help them with their gut microbiome, for example.
So again, I would encourage you to reach out with a description of the product and ask specifically what applies, because there's always nuance in that I just can't answer
Thank you.
accurately.
Yeah, your question. I think so. You keep going, yeah.
Irina Chiripanova. For the phage being developed for drug-resistant bacteria treatment in human, could you comment on extent of animal studies in non-clinical sort of area prior to first in human?
So you're getting at whether there are any specific expectations for types of animal studies.
The animal
Yeah.
model testing.
So for the products that we regulate in OVRR, we do not have a specific requirement for preclinical animal studies. A lot of sponsors find that there are animal models that are useful to them for development purposes to understand the pharmacology. We don't require traditional GLP tox. But there are scenarios where safety evaluations that may be needed depending on the details of the product and how it's being used. The details of that would, and I would say a pre-IND is a great place to ask about the details that might apply to your product. And those may or may not be animal studies, depending, again, on the details.
Thank you.
I'm going to check quickly to see if there are any questions online. Go ahead.
So the first online question is, are there any specific types of infection that would be more suitable for phage therapy?
That's not a question that I can really answer. It was a little bit quiet in the room. The question was whether there are any particular types of bacterial infections that would be more suitable. That is up to sponsors to decide what they feel their product is best suited for.
We have a second question. So for a product development IND, the bacterial strains that cause a particular disease can shift over time. Is it possible for sponsors to change the phages in their product at any point in the development process?
And so the question was whether it's possible or permissible to change the phages that are in a product in a particular within an IND. And so the answer is yes, but if the way we sometimes see this question is a product has 3 phages and someone wants to swap out one of the phages, or they want to add a phage and make it a 4-phage product. As long as the extent of the change is within a certain range, it can be acceptable to make that change to the product composition within an existing IND. There are some reasons when it might not be okay, but it is potentially possible to make that change within an existing IND. I would caution people, however, that making those kinds of changes later in your product development process may complicate that substantially, because as you're moving into phase 2 and phase 3, you are looking to develop safety and effectiveness data based on your final product. If you make a change too late, you may end up having to repeat a bunch of work. So it can be possible to change that composition. The earlier you do it, the better, and you should think through the potential consequences in terms of bridging any early data that you may have.
Next question. For an expanded access IND, you mentioned that the CMC information requirements are more flexible. Can you elaborate more on what is expected for those single patient INDs?
Yeah, so one example of some of the information that is a little more flexible in those cases would be information about the manufacturing host. There are sometimes cases where the phage that's being used may actually be grown on an isolate that was collected from the patient, a bacterial isolate that was collected from the patient. And so in that case, you may not have full sequencing, you may not have, you're growing it on something that you know is a pathogen, and so we're going to rely more on the information that you have about purification and final testing before putting it back into the same patient. That is a scenario where we would have a lot less information about the host itself and a lot less control over, you haven't taken the time to engineer out undesirable genes from that host, but in the context of that specific patient's need and their existing infection, it might be considered a reasonable benefit risk profile.
The last question is a very general question. Does the difference in CMC requirements mean that participants in phase 1 trials are exposed to more risk?
So I don't think that's an accurate way to look at it. And I think there's a good explanation of this in the guidance itself, where it talks more about the fact that the level of the way that you control quality and and limit risk when you are at a very early stage and a very small scale of manufacturing can take a different form. So we can protect individual study participant safety to a similar degree with different types of process controls at a very small scale versus at a future commercial scale.
I was just going to say that, thank you so much, Susan. I mean, this is a great presentation. And I really like the fact that at the end, amongst your takeaways, you mentioned communication and a constant communication in terms of just engaging with us. And we have experts here, right, who can really answer your questions based on your product profile. And I know we didn't completely address a lot of the questions because we didn't have product specific details, and this is not the forum for that, but clearly communication is key. So thank you so much, Susan Lehman. All right, so now we move on to session 8, and I think we moved on. Okay, so great. So this session 8 is about regulatory considerations in the safety assessment of vaccine adjuvants and adjuvanted vaccines. Dr. Meghan Maguire Thon is our presenter today. She's a regulatory project manager. She's a team lead in the Office of Vaccines. And in addition to this, she serves as co-chair and representative on process improvement committees within our office and our center. I will stop with this brief introduction because I know this is a very exciting talk, and I'll turn it over to Dr. Meghan Maguire Thon.
Thank you so much, Dr. Malik. I really appreciate it. Okay, so Let's get started. So today I'd like to start with a little bit of a background on vaccine adjuvants and their use in preventative and therapeutic vaccines, specifically for infectious disease indications. I'll then present an overview of some of the adjuvants that can be found in licensed vaccines in the United States. And finally, we'll walk through some of the regulatory considerations for non-clinical and clinical safety assessment of investigational vaccines containing novel adjuvants. The Office of Vaccine Research and Review, or OVRR, which is where I belong, regulates vaccines specifically for infectious disease indications. These can include preventative vaccines that are intended to prevent infectious disease by inducing a protective immune response, and they can also include therapeutic vaccines that are intended to treat infectious diseases like HIV. This would be in contrast to something like a therapeutic vaccine for a cancer indication, which would be regulated by the Office of Therapeutic Products, or OTP. So what exactly is an adjuvant? An adjuvant is a substance that's added to a vaccine antigen to enhance and modulate the immunogenicity of that antigen. Simply put, adjuvants are it's important to note, in addition, that adjuvants are not licensed as a standalone product. It's that specific antigen and adjuvant formulation together that is carried through clinical studies and then ultimately submitted for licensure. Adjuvants can be added to antigens and to vaccines for several different reasons. This can include increasing the immune response. It can include antigen sparing, decreasing the dose or the frequency of doses, and also increasing that immune response in a particularly vulnerable population. There are several adjuvants that are included and can be found in vaccines that are licensed in the U.S. Overall, there are aluminum salts that can be found in many vaccines. Arapanrix includes ASO3 and oil and water emulsion. Fluad and Audens include MF59, a squalene-based oil and water emulsion. Shingrix is formulated with ASO1B, monophospholipid A, or MPL, and QS21 combined in a liposomal formulation. And Arexvy contains ASO1E, which contains half the amount of MPL and QS21 as in ASO1B, but otherwise the same formulation. Cervarix contains ASO4, MPL, and aluminum hydroxide. And Nuvaxovid is formulated with Matrix M. Cyfendus and Heplisav-B are formulated with CpG 7909 and CpG 1018 respectively. So now I'd like to turn to vaccine safety, which is paramount and a key consideration of everything that we do here at FDA. Under Section 351 of the Public Health Service Act, vaccines are licensed based on the demonstration of safety, purity, and potency, and that the manufacturing facility and process it upholds those same standards. Simply put, vaccines that are demonstrated to be safe and effective, and those that can be manufactured in a consistent manner, are licensed by the FDA. So I think it's important to take a second here to look at what safety means in a regulatory framework. Safety, as defined by the Federal Code of Regulations 21 CFR 600.3, is the relative freedom from harmful effect to persons affected directly or indirectly by a product when prudently administered, taking into consideration the character of the product in relation to the condition of the recipient at that time. This is really important as it implies that benefit risk assessment. So as we think about the clinical development of a product, we've sort of talked about this a little bit this morning, but we progress from preclinical studies through a small phase 1 study that could include approximately 20 to 80 participants to a larger phase 2 study that could include up to hundreds of participants, and then further to a phase 3 study that could include thousands of participants. And safety remains a paramount concern and primary endpoint in all of those phases. I think it's also important to note here that vaccines are slightly different. They're largely given to healthy populations. And then as we take a slight shift and look at the review and regulation from an FDA perspective, we can again begin to discuss these products in the pre-IND stage, and we certainly encourage that. And then as a product moves through those same phase 1, phase 2, and phase 3 studies, we continue to gather safety and effectiveness and efficacy data on those products. The totality of that data, including all of the safety data that's been collected, can be submitted to a biologics license application or a BLA. But our safety analysis doesn't stop there. It continues through phase 4 and other post-marketing monitoring. So when we think about an IND submission, we're going to look at the safety of a product from many different angles. We're going to look at the product testing and characterization, the non-clinical safety evaluation, and the clinical evaluation. So first, I'd like to start with the product testing and characterization. From a regulatory framework, the goals of, we often refer to the Code of Federal Regulations for guidance and what should be included in an IND submission. Particularly, we're looking to 21 CFR 312 for information on chemistry, manufacturing, and controls. We're looking at 21 CFR 610 eventually for lot release, potency, sterility, purity, and identity information. And finally, for a product that contains a novel adjuvant or an adjuvant alone, sorry, not an adjuvant by itself, but a product that contains an adjuvant. We're going to refer to 21 CFR 610.15 on constituent materials. Adjuvants are considered constituent materials, such that all of the ingredients shall meet generally accepted standards of purity and quality, and an adjuvant shall not be introduced into a product unless there's satisfactory evidence that it does not adversely affect the safety and potency of this product. So again, we come back to the safety of the product and its benefit risk assessment. So when we think about submitting an IND, we're certainly looking for a complete package for the chemistry, manufacturing, and controls that includes not only the antigen and the adjuvant, but also that adjuvanted vaccine formulation together. This could include details on the raw materials, purification, identity, potency, and sterility. It should likely include product-specific tests, and it should also include lot release and stability data as available. Particularly when we're talking about an adjuvant, this could include information on the identity, the content, the degree of adsorption and particle size distribution, and also the stability of that adsorption or emulsion. Submission of an IND that involves the use of an adjuvant should also include a rationale for the use of the antigen and that particular adjuvant. Should also include a rationale that provides information on the choice of dose and the ratio of adjuvant to antigen, and how that is planned to be, how that your choice of those came to be. We also encourage manufacturers and sponsors to characterize the immune response to those adjuvanted vaccines. So next, I'd like to move to some of the non-clinical safety evaluation. Again, thinking from a regulatory perspective, we refer to the Code of Federal Regulations, 21 CFR 312, such that an IND submission should contain adequate information about the pharmacological and toxicological studies on the basis of which the sponsor has concluded that it is reasonably safe to conduct the clinical investigation. So simply put, the safety of that adjuvanted formulation should be demonstrated in adequate and well-controlled preclinical licensure study or pre-licensure safety studies. And really the goal is to establish a safe and immunogenic dose and any potential toxicities. So we can start with some of the pharmacology studies, and those may evaluate some product-specific concerns. Sponsors are encouraged to conduct in vivo proof of concept studies that demonstrate the immune response to an antigen, and those can be with and without the adjuvant. And again, if an animal species is sensitive to that antigen and adjuvant, then the animal model may be useful for conducting some of those pilot studies and inform the dose ranging and to demonstrate an added benefit of the adjuvant. Moving on to some of our toxicology studies, a toxicology study that's submitted to an IND should be conducted in compliance with good laboratory practices, and it can help in establishing a safe and immunogenic dose prior to those phase 1 clinical trials. And it can also help identify potential local and systemic adverse events. Sponsors may also consider referring to the WHO guidelines for additional information on novel adjuvants and toxicology studies. But ideally, when we're looking at and reviewing a toxicology study, it's ideal that the lots that are used in that toxicology study are manufactured in the same process and formulation that's ultimately going to be used in that clinical trial. So next, I'll move to the clinical evaluation. So there's several considerations that go into play when designing a clinical study, including the dose selection of that antigen and adjuvant. But we really want to apply a consistent approach to safety monitoring, and that can include monitoring of adverse events of special interest, or AESIs, and these largely focus on autoimmune and neuroinflammatory diseases. And I'll speak more about these shortly. With a phase 1 study, this may represent the initial use of an investigational vaccine in humans. Again, as we stated before, these phase 1 studies are typically limited in number. They typically include approximately 20 to 80 healthy participants. And for adjuvanted vaccines, we often see a non-adjuvanted control arm and a placebo arm, and we find that those controls can be useful. However, it is important to note that we do not typically recommend an adjuvant alone arm because the placebo provides a more meaningful comparator for safety, and an adjuvant alone arm may run the risk of reactogenicity. So again, going back to that benefit risk profile. While the dose selection of an adjuvant is generally left to the discretion of the manufacturer, there are some considerations. Using a low adjuvant dose may carry the risk of suboptimal dosing, whereas in contrast, a high adjuvant dose may carry the risk for choosing an adjuvant and dosing that's higher than optimal, which could lead to increased reactogenicity. But ultimately, the safety and immunogenicity of that adjuvanted vaccine should be assessed in the clinical trial. And early clinical trials may also provide a demonstration for the added benefit of the adjuvant. So let's talk a little bit more about the demonstration of the added benefit of the adjuvant. So as we said before, the rationale for the use of the adjuvant and the dose selection should be provided in the IND submission. And that added benefit can be supported in several different ways. This can include the non-clinical studies, and in vitro or in vivo proof of concept studies in an animal model, and that those could be conducted prior to the use in the clinical study. Early clinical immunogenicity studies can provide the rationale and some evidence of added benefit of an adjuvant by comparing that adjuvanted vaccine versus the unadjuvanted vaccine. And some of the endpoints and outlooks of that kind of trial could be an enhanced immune response or antigen sparing or other options. And then supportive data could also come from the use of that adjuvant with related vaccine antigens, so that all of that data in totality can provide a rationale and the demonstration of added benefit. So again, if possible and if available, we encourage sponsors to provide information about the mechanism of action of the adjuvant as well. Because adjuvants are not considered active ingredients from a regulatory perspective, I do want to note here that manufacturers are not required to demonstrate the added benefit in a phase 3 efficacy study, though. So typically, as we said, these studies providing an added benefit are conducted in early phase studies, not a phase 3 trial that would compare a vaccine antigen with and without the adjuvant. However, additional studies may be requested by the agency on a case-by-case basis, and this could include when safety concerns are identified with the adjuvant or when the sponsor intends to claim superiority of the adjuvanted product over an unadjuvanted vaccine. So as you develop and as we review a clinical study, one of the things that we're really focused on is the safety monitoring. And in fact, for a product that includes a novel adjuvant or one that is an investigational product, the safety monitoring is generally really similar. We request the sponsors follow solicited adverse events for at least 1 week after each vaccination. We request the solicited adverse events are followed for at least 4 weeks after vaccination, and serious adverse events and death are followed for the duration of the study. However, when we do see a product with a novel adjuvant, we are looking for an extended duration of safety follow-up, and this would include up to 12 months following the last vaccination. And this could include monitoring for vital status, serious adverse events, death, new onset medical conditions, and those adverse events of special interest, or AESIs. And typically, those focus on the autoimmune and neuroinflammatory diseases, and we typically request that the MEDRA preferred term that's included in the immune-mediated and autoimmune disorders of the standard MEDRA query, or SMQ, are employed. Some of the examples of AESIs include neuroinflammatory disorders, musculoskeletal and connective tissue diseases, and also gastrointestinal disorders, though this list is not exhaustive. So just to kind of recap a little bit on the safety evaluation, I'd like to recap that there's no regulatory requirement to evaluate the safety of an adjuvanted and an unadjuvanted vaccine in a phase 3 study. And when providing that rationale for selection of the adjuvant and the added benefit of that adjuvant, the safety experience with that same adjuvant formulated with other antigens can contribute to the safety evaluation of that adjuvant, as well as when information is submitted to a biologics license application, safety experience that can be obtained from domestic or foreign clinical studies. So just as an overview to kind of walk through what we've discussed today, we've talked about vaccines and vaccine adjuvants. I want to point out again that vaccines are considered constituent materials according to the regulations. And when submitting an IND submission, manufacturers should provide that justification, including the adjuvant in the vaccine formulation, and that could include some of that added benefit information. We also want to see in our IND submissions all of that complete CMC information, including the testing and characterization of the antigen, the adjuvant, and that formulated adjuvanted vaccine. As we discussed several times, the demonstration of added benefit could come from the non-clinical assessments. It can also come from those early phase clinical immunogenicity studies. And then finally, I'd like to leave you with one last thought and quote that as the FDA's experience of adjuvanted vaccines, this dialogue is something that we take very seriously. I'd like to thank the many colleagues who have contributed to this presentation and my learning experience. So again, I'd like to thank all of you, and I'd be happy to take any questions.
Hi, Lauren Sugar-Mart, CIA LBG. Just a quick question regarding can you speak to the utilization of more than one adjuvant in a drug product in terms of the requirements from a quality perspective and a clinical perspective? I think that unusual question. Yeah, thank you so much for that question. But I think that, again, I would encourage you to come in with a pre-IND and we can, focus specifically on some of those product questions. I'd like to also point out that when you come in with an IND, I think that, we'd certainly look at all of the information that's provided, all of that CMC information, the plan for the clinical study. And it's really, I guess, helpful from the regulatory perspective that if you come in with all of that information to start with, then it can all, if agreed, then we can evaluate it all under a single IND rather than adding in different products at a later time, which could complicate your clinical study. Thank you.
I think we have a question online. Do we have questions online? Yeah, we can start with those, yeah.
Yeah. The first online question is, are the lipid nanoparticles in mRNA vaccines treated as adjuvants?
No, thank you so much for that question. No, lipid nanoparticles are not considered adjuvants at this time.
Second question, what would be the minimum number of participants who received the investigational vaccine during clinical development to be considered a sufficient safety database to demonstrate safety and allow licensure of a vaccine?
Thank you for that. Yeah, that's a really good question. I think that it certainly could be a case-by-case basis, but we certainly look for some of those early clinical studies. We encourage sponsors to consider sentinel groups, which would administer the vaccine to a small number of individuals at first, and look at those safety profiles, and then increase to larger populations. And as you progress to phase 3, I think we would generally like to see about 3,000 participants.
And many of these obviously depend upon the product profile and safety considerations that are very specific to the product, as was mentioned in the slides. Yeah.
Next question, is there a list of non-novel adjuvants versus novel ones?
Super novel.
No, thank you. That's a really interesting question. There are certainly adjuvants that are present in licensed vaccines that we do have some experience with, but I don't believe that we have a published list of novel adjuvants versus non-novel adjuvants. I think we would encourage sponsors to take an approach that monitors safety appropriately, and we would certainly help guide sponsors through our interactions in a pre-IND and through the IND stages.
Next question, how does the FDA suggest sponsors assess the starting dose of the adjuvant?
Thanks, that's a really good question as well. So as I mentioned, that demonstration of added benefit of the adjuvant, the adjuvant to the antigen can be assessed in both non-clinical studies, early phase clinical studies. So there's some, there could be some literature references that can help, I guess, start the non-clinical studies, but we would encourage sponsors to come in for pre-INDs and discuss with an in vivo model that could be sensitive to that antigen and adjuvant combination. And then some of those preclinical studies can start to look at some of the dose escalation and combinations that would provide for something like antigen sparing or an increased immune response.
Next question, how and when are sponsors required to evaluate clinical benefit of an adjuvant?
Oh. Thank you. Yeah, so we typically would like to see the added benefit of an adjuvant, and that rationale can be provided through several different mechanisms, and it can start with those non-clinical studies. It can be done in some of the early phase clinical studies, and those would typically be an unadjuvanted and an adjuvanted vaccine, and those would typically not include an adjuvant alone arm, as that could pose a risk without an added benefit. But again, we would not request in general, except for a case-by-case basis, we would not request a phase 3 clinical study that evaluates the adjuvanted and unadjuvanted vaccine that in order to provide that rationale for added benefit.
Can you expand on the rationale for monitoring of AESIs in clinical studies?
Thank you. Yeah, so we recognize that the occurrence of some of these adverse events of special interest are rare and could possibly be theoretical, but we really strive to systematically assess the risk of all vaccines that contain novel adjuvants, and we want to identify any potential safety concerns as they arise. And as you start to increase the participant numbers in advanced clinical phases, then we want to maintain the safety profile and assess that risk.
Our next online question. If you do not consider LMPs as an adjuvant, then they are considered as a formulation excipient. Is that correct? Other agencies such as EMA have a different approach. How do you harmonize that for a global vaccine development plan?
Yeah, thank you. Yeah, I would have to defer to some of my colleagues on the lipid nanoparticles, unfortunately. But I do think that, we evaluate the totality of information that we have included in a biologics license application for a particular product, and it's all independently reviewed, and our reviewers spend a lot of time assessing that information. So I think that in terms of products that are, approved elsewhere, we take a very serious approach to safety and do perform our own independent review.
That's it for online questions. Thank you.
I was going to ask if there are any questions in the room. I see someone walking up, so let's just give it 1 second before. Yeah, do you have a question? Yes, please.
I don't have a question. This is Sudhakar. Just on the previous question, when Meghan deferred to the colleagues, in terms of interactions with the national regulatory agencies, I think we do periodically interact with the national regulatory agencies, EMA, FDA, Health Canada, et cetera. And then we do discuss specific issues in the context of safety related to like lipid nanoparticles, et cetera. But that would be on a case-by-case basis under confidentiality. So we share perspectives, but that does not factor in terms of our decision-making. Thank you.
That was helpful addition, and hopefully that helps clarify the situation in terms of how we look at product attributes, including the LNP component, and clearly it's not an adjuvant. I think that was the takeaway message for today. Any other questions in the room or online? Right. With that, we can adjourn early. And thank you so much, Meghan. This was a great talk. And I think it was really satisfying given the number of questions we got. So thank you.
So unusual for a conference, and we are actually done early rather than behind schedule. So you've earned yourself a nice long break. We will definitely be starting up at 10:50. So that back here in the room, and for those of you on Adobe and following us on YouTube, we will start right at 10:50. With the next session from the Office of Therapeutic Products. So thank you very much.
Good morning, everyone. My name is Zehra Tosun. I'm the branch chief at the Tissue Engineering branch at the Office of Therapeutic Products. I will be moderating the next session, and joining me as our online moderator, Timothy Kamaldenov, who's in the back of the room. And we are pleased to have two distinguished colleagues presenting today. And before we get started, I appreciate if you can silence your cell phones. Let's get started. So our first presenter is Nadia Whitt. And Ms. Whitt serves as the branch chief in the Office of Review Management and Regulatory Review within the Office of Therapeutic Products at CBER. And she held this role since 2024. And prior to this position, she served as the lead regulatory project manager, where she trained new regulatory project managers and provided regulatory expertise to her team. And she has reviewed and led multiple application types, including BLAs, INDs, IDEs, and 510Ks, while serving as the primary point of contact between FDA and sponsors. Please join me welcoming Nadia Whitt.
Hi, good morning. Sorry. Thanks, Sarah. Yeah, my name is Nadia Whitt. I'm a branch chief in the Office of Regulatory Management and Regulatory Review in the Office of Therapeutic Products at CBER. And today I will be presenting the highlights and frequently asked questions in the Office of Therapeutic Products. Before we dive into the specifics of working with OTP, I want to provide important context about our organizational evolution. By 2022, cell and gene therapy development was accelerating exponentially. The field was experiencing unprecedented growth. CAR T therapies, gene editing technologies, novel viral vectors. Our existing structure at OTAT, the Office of Tissues and Advanced Therapies, was no longer adequate to support the growth trajectory. On February 26, 2023, we transformed from OTAT into the Office of Therapeutic Products, OTP, a super office specifically designed for the future of advanced therapies. The scope of this transformation was substantial. We expanded from 5 divisions to 6 specialized offices. These 6 offices now oversee 14 divisions, which comprise 33 branches with focused expertise across the product lifecycle. This reorganization improved discipline alignment across review teams, increased our review capacity to handle the growing workload, and enhanced our expertise in cell and gene therapies. The result is better timeliness and consistency in our interactions with sponsors. OTP was built for the future of cell and gene therapy, and that future is now. Today, I'll show you how to navigate this structure effectively to support the development of your programs. Here's our agenda for the next 30 minutes. We'll start with OTP structure and the products we review. Then we'll focus on best practices, practical strategies for successful submissions, and effective communications with your regulatory project manager. We'll address frequently asked questions and finish with key resources and contacts. So let's begin with how OTP is organized. OTP has 6 specialized offices organized to align disciplines and product types. 3 of these offices focus on CMC. We have cellular therapy in human tissue, gene therapy, and plasma protein therapeutics. Now let me emphasize our clinical review capacity. The Office of Clinical Evaluation is significantly large, comprising 3 divisions with 8 branches total. Here's an important distinction between OND CDER and OTP CBER. CBER's of Office New Drugs is structured by disease area, whereas CBER's OTP is structured by the products regulated in each office. Why does this matter? Advanced therapy products often cross multiple organ systems and therapeutic areas. A gene therapy platform might treat neurologic, metabolic, and ophthalmologic diseases. A CAR T platform might be used for different types of cancer. OTP's product-based structure allows this flexibility while maintaining deep therapeutic expertise in general medicine, oncology, and hematology. The Office of Pharmacology and Toxicology evaluates preclinical safety, And OR/MRR is where your RPM is located, coordinating across all 6 offices. Another important distinction between O&D and OTP, in OTP, you interact with 1 RPM for all aspects of your file, whereas in O&D, you interact with 2 RPMs, 1 OPQ RPM and 1 CMC for CMC-related matters, and 1 O&D RPM for all other issues related to your file. The key point, the OTP structure provides specialized expertise across CMC, clinical, and pharmacology toxicology disciplines. Each office has the depth needed to handle the complexity of advanced therapies. And here's how that coordination works. When you submit, your submission arrives at CBER's Document Control Center, the DCC. They process it and send it to OR/MRR for initial triage. OR/MRR assigns a regulatory project review manager, your RPM, who becomes your primary contact throughout the entire review process. Your RPM then assembles a multidisciplinary review team based on your product type. This typically includes a CMC reviewer from gene therapy, cell therapy, or plasma protein therapeutics, a pharmacology toxicology reviewer who evaluates your preclinical safety data, and a clinical reviewer from general medicine, oncology, or hematology. We also bring in biostatistics, bioinformatics, and clinical pharmacology, as well as any consults that are needed. Here's the key. Your RPM coordinates all of this. Your RPM assembles the team, schedules meetings, sends information requests, manages timelines, and communicates the final decision. One contact, coordinated review, that's how OTP works. If you have any questions, contact your RPM, or if you're not sure who your RPM is, contact OTP RPMS. Now let's talk about the diverse products we review. What makes OTP unique is the extraordinary diversity of our portfolio. We don't just review one type of product or one therapeutic area. We review products that span completely different technology platforms, manufacturing approaches, and therapeutic applications. To give you a sense of this diversity, let me highlight what we approved just in 2025. We approved 5 gene therapies, each using different approaches for different diseases: Wiscara for an immune disorder, Evisma for a neuromuscular disease, Papzymos for a respiratory condition, Zavaskin for a skin disorder, and Inselzo for an eye condition. 5 completely different therapeutic areas, all using gene therapy platforms. We approved blood and plasma products, Facility, a fibrinogen product, and Qvigi, an immune globulin. We approved Advanced Nerve Graft, a tissue-engineered product that's literally rebuilding nerves. That's 8 products in 2025 across 3 different technology platforms and multiple therapeutic areas. And these are just examples from 1 year. As you can see here, we review many product categories: gene therapies, including viral vectors and gene editing, cellular therapies from CAR T to stem cells, therapeutic vaccines and immunotherapies, blood and plasma products, tissue-engineered products, combination products, and xenotransplantation. The point is this: OTP 6 specialized offices were designed specifically to handle this extraordinary diversity, from the newest gene editing technology to well-established plasma fractionation. We have reviewers with deep expertise in every platform and every therapeutic area. Not sure if OTP reviews your product? You can contact CBER product jurisdiction. Now let's talk about best practices to work with us effectively. Best practice number 1, and this is critical: engage early and often. Why does early engagement matter? 3 reasons. First, you can align on your regulatory strategy before making major investments. Second, you identify potential issues early when they're easier to address. Third, you understand exactly what data you'll need for your IND and eventually your BLA. We offer 2 types of early meetings: Interact and Pre-IND, and let me help you when you need to use each one. Interact meetings are for very early stage development, before you've designed definitive toxicology studies. You should have your product identified and some preliminary proof of concept data, but your manufacturing process may still be in development. These are exploratory discussions. The meeting happens within 75 days, and importantly, you must submit your meeting package with your request. There are no formal meeting minutes for the INTERACT meetings. Pre-IND meetings are for the IND enabling stage. By this point, you should have your manufacturing process defined, your assays developed, and your proof of concept studies complete. You're ready to discuss the definitive studies needed for your IND. The meeting happens within 60 days. Your package is due 30 days before the meeting, and you'll receive formal meeting minutes within 30 days. Here's the critical point. OTP grants only one INTERACT meeting and only one pre-IND meeting. That means both must be comprehensive and multidisciplinary, covering clinical, CMC, and pharmacology toxicology. If your questions are not multidisciplinary, we may deny the meeting. Can you have both an INTERACT and a pre-IND meeting? Yes. INTERACT first for early guidance, then pre-IND when you're ready for those IND-enabling discussions. And a pro tip is don't wait until you think you have all the answers. That's often too late. Best practice number 2, know where and how to submit. This slide has all the email addresses you need, and I won't read through each one. Instead, let me highlight the key distinctions you need to understand. First, meeting requests versus IND submissions are handled differently. For INTERACT and pre-IND meeting requests, you can submit via email as PDF attachments. ECTD is encouraged but not required. This makes it easier for you to get early feedback without the technical burden of ECTD formatting. For IND submissions, it depends on whether you're commercial or research. Commercial sponsors must use the Electronic Submission Gateway, ESG, with ECTD format. Research sponsors have more flexibility and can email their submissions, although we do prefer ECTD. What is ESG? It's FDA's secure web-based portal for submitting regulatory applications electronically. Commercial sponsors use ESG to submit INDs, BLAs, and all related documents in ECTD format. If you have questions about ESG or ECTD submissions, contact eSubPrep. Next, I want to bring your attention to secure email. Make sure that you set up secure email before your first submission. If you haven't set up secure email yet, you can contact secure email at FDA before you submit anything. This is a requirement, and not having it set up can delay your acknowledgement letter and communications from your RPM. It's a simple step, and it just takes a few minutes, but it prevents a lot of delays later. Third, expanded access has specific pathways. For non-emergency single-patient INDs, there's a standard email process with specific form requirements. For emergency INDs, you email directly to our regulatory project management staff, and if it's after hours, after 4:30 on weekdays or all day on weekends and holidays, call the FDA Emergency Call Center. The email address and references are all on this slide for your reference. The key is understanding which pathways applies to your situation. If you're ever unsure, just contact OTP RPMS and we'll help you figure it out. And just a pro tip, please make sure to set up your ESG account and secure email before you need them to save you a lot of time. All right, best practice number 3, prepare quality submissions. First, timing matters. You might consider submitting Monday through Wednesday and not on Fridays. This maximizes the review time within the 30-day clock and ensures your RPM is available if questions arise. Make sure your authorized contacts are available throughout the 30-day review period and have 1 or 2 backups in case anyone's out. Second, your cover letter is critical. Each submission should include a clear cover letter with your product title and not just the company moniker like BLD 105. We do want a description of the product. And you should also include the indication, sponsor name, complete address, all authorized representatives with accurate contact information, including phone number and email. Any cross-referencing information, including IND or master file numbers with the letter of authorization included, previous OTP interactions such as INTERACT or pre-IND meetings with the pre-submission number, and that's the PTS number, a clear outline of what the submission includes, like if you've requested a fast track designation, and for amendments, the submission number in the subject line should be included. Third, for meeting packages, prepare clear focus questions grouped by discipline, like clinical, CMC, farm talks. Provide context for each question, include critical supporting documents like key publications or data tables, but don't overwhelm us with unnecessary detail. Limit your questions to about 10, and that includes sub-questions. Quality over quantity. Focus questions get better answers. And for pre-IND meetings specifically, remember your questions must be multidisciplinary, covering clinical, CMC, and farm talks. Here's what makes a good question. Don't ask, what do you need for my IND? That's too vague. Instead, try something like, we propose a 3-month GLP toxicology study in non-human primates at this dose and schedule. Is this adequate to support our phase 1 trial in this patient population? Be specific about your approach and what you're trying to support. Another pro tip here, a complete, well-organized submission on Monday gets more review time than a rushed submission on Friday, so please plan ahead. Best practice number 4, how to communicate effectively with your RPM. Here are some do's for effective communication. Do use secure email for all regulatory communications. Include your pre-submission number, IND or BLA number in the subject line, so it's easy to track. Provide context and specific questions. Don't just ask, what's the status of my IND? Respond promptly to information requests. Check your acknowledgement letter for your RPM's contact info, and that should be sent to you within 2 weeks of an original submission. And be patient. RPMs are managing multiple complex submissions simultaneously. To give you a sense of that workload, OTP receives an average from the past year of 14 new INDs per week. And that's just the original INDs. RPMs are also handling amendments to existing INDs, meeting requests, BLA submissions, annual reports, safety reports, and all the communications that go with this. And these are just examples. And additionally, RPMs attend regular training to keep up with updates and regulations, guidance documents, and SOPs, so they can effectively manage your submission with the most current information. So when you reach out, understand that your RPM is juggling many priorities while staying current on regulatory requirements. Here are some don'ts. Don't send the same question to multiple email addresses hoping for a faster response. That actually slows things down and creates confusion. Don't expect immediate responses to non-urgent questions. Don't forget to update your contact information when it changes. And don't submit inquiries to CBER DCC email. That's for submissions only. They get confused when you ask them questions. So please send your questions to RPMs, to your RPM or to OTP RPMS. And don't contact reviewers directly. Always work with your RPM. Remember, your RPM knows the status of your submission from a multidisciplinary perspective. They're coordinating across CMC, clinical, and farm talks, and going directly to a reviewer bypasses that coordination and can only create confusion. For time-critical issues, mark your email subject line time-sensitive with your submission number. Provide a clear deadline and rationale for why it's urgent. If you don't get a response from your RPM within 48 hours, contact OTP RPMS. The pro tip here says it all. Clear, organized communication helps your RPM help you more effectively. One contact, coordinated review. Your RPM is your partner in navigating the regulatory process. Now let's address some of your frequently asked questions. When does my research IND become commercial? This is a critical question because it has significant implications for how you submit. Your IND becomes commercial when you plan to submit a pivotal clinical investigation to support a future marketing application or when you intend to commercialize the product. When this happens, you have 4 required actions in this order. First, set up an ESG account. Second, switch to ECD format for all future submissions. Third, select commercial IND on your Form 1571 Field 7B. And lastly, notify FDA in your cover letter that your IND is now commercial. There's one exception. If you're phase 2 or phase 3, if you're a research IND and your phase 2 or phase 3 protocol is solely for research purposes and not intended to support commercialization, you may submit a justification in your cover letter. If FDA agrees with your justification, your IND can remain classified as research and ECTD requirements won't apply. All right, FAQ number 2. When should I request end of phase 2 and pre-BLA meetings? These are critical milestone meetings and timing is important. End of phase 2 meetings should be requested after your phase 2 data is available, but before you start phase 3. The purpose is to align on your phase 3 trial design, endpoints, and statistical approach. This is your opportunity to ensure FDA agrees with your pivotal trial design before you make that major investment. Request well in advance of your planned phase 3 start. Pre-BLA meetings should be requested 3 to 6 months before your planned BLA submission. By this time, your pivotal data should be available and your BLA should be substantially developed. The purpose is to discuss BLA content and format, address any outstanding issues, and align on labeling concepts. This meeting helps ensure your BLA submission will be complete and addresses any remaining questions before you submit. FAQ number 3, can I cross-reference another IND or master file? This is a very common question, and the answer depends on the status of your IND that you're cross-referencing. For active INDs and master files, you can cross-reference. You'll need a letter of authorization or an LOA from the original IND or master file holder, and they also have to submit an amendment on their end with the letter. You must reference the original source directly, no intermediate sources, so you can't cross-reference an IND that's cross-referencing another IND. It has to be the immediate source, and include the LOA in your submission. For inactive INDs, you can also cross-reference as long as the cross-referenced information is up to date. You'll still need an LOA from the original IND holder. And for withdrawn INDs, you cannot cross-reference. You must submit complete sections. If you know the IND you're cross-referencing will be withdrawn, submit your complete data before the withdrawal happens. That's an important caveat for all cross-referencing. The LOA can be withdrawn at any time by the sponsor. And if that happens, you'll need to provide the information by other means or your IND may go in clinical hold. Key message here, active and inactive INDs can be cross-referenced with proper authorization, but withdrawn INDs cannot. FAQ number 4, how do I submit an N of 1 IND? N of 1 INDs are for individualized therapies. For example, the first application of gene editing as a treatment in a new therapeutic area or a patient-specific gene therapy. We have 2 recommendations depending on your timeline and the complexity of your questions. If time permits, we recommend you request a pre-IND meeting with FDA for feedback prior to submitting your IND. This allows you to discuss your approach, the preclinical data needed, and the clinical trial design. Email your meeting request to CBER DCC. For a more accelerated timeline, you can submit a Type D meeting request with FDA. Type D meetings are scheduled within 50 days and are appropriate when you have 1 to 2 focused topics with 3 to 5 questions total. Because the meeting background package is included with a Type D meeting request, it allows for an expedited FDA response, and you'll get a decision on whether the meeting is granted within 14 days. For N of 1 INDs, particularly those involving gene editing or novel approaches, we refer you to these 2 guidance documents listed here. FAQ number 5, how do I know if my study qualifies for an IND exemption? This is an important question because conducting a study that requires an IND without one can have serious regulatory consequences. First, refer to this FDA guidance here. This guidance outlines the specific criteria for IND exemption. If you're unsure whether your study qualifies for an exemption, here's what we recommend. Submit an IND for review and include your rationale for IND exemption in the cover letter of the submission. The FDA review team will evaluate your justification and protocol to determine whether an IND is actually needed. It's better to submit an IND and have FDA confirm the exemption than to proceed without an IND when one is required. This protects both you and the study participants and ensures you're in compliance with regulations. FAQ number 6, do I need to respond to Interact and pre-IND meeting comments? This one's really important, and the answer is yes for both meeting types. For Interact meetings, you should submit responses to the FDA comments, but the key is you don't submit it back to the Interact meeting request. Instead, you include the responses in your future IND submission or your future meeting request. For example, if you have a pre-IND meeting request, after your Interact, include your Interact responses with a point-by-point response in that pre-IND meeting package. For pre-IND meetings, you'll receive formal meeting minutes within 30 days, and you should submit the responses to those comments in your IND submission when you're ready to submit. Again, please provide the point-by-point response to our comments. And why does this matter? It demonstrates that you understood FDA's advice, clarifies any misunderstandings early, and shows how you addressed FDA's recommendations in your development program. This creates a clear regulatory record that can be referenced throughout your review. Okay, my last frequently asked question, and this one is about safety reporting requirements for commercial INDs. As a reminder, as of April 1st, 2026, which is now in effect, all commercial IND sponsors must submit individual case reports, ICSRs, for serious and unexpected suspected adverse reactions electronically in ICH E2B R3 format. This is required by Regulation 21 CFR 312.32. This requirement applies to all commercial sponsors regardless of trial size or organizational resources. And you have two compliant pathways. First, the FDA Patient Safety Report Portal, the PSR portal. This allows manual entry of safety reports through a web-based interface and is recommended for smaller sponsors or those without extensive technical infrastructure. Second, the Electronic Submission Gateway, ESG, where you submit E2B R3 compliant XML files directly. This is better if you have the technical capability. Paper MedWatch forms are no longer accepted for commercial INDs, and if you're a commercial sponsor and you haven't registered yet, you can contact eSubPrep. Okay, so a few key takeaways. Let me summarize what I've just presented. First, OTP was built for you. Our reorganization into 6 offices, 14 divisions, and 33 branches was designed to match the innovation happening in cell and gene therapy. We have enhanced capacity and specialized expertise with the same commitment to innovation, but better equipped to serve you. Second, engage early. Use Interact meetings for exploratory discussions and pre-IND meetings for IND-enabling plans. Remember, OTP grants only one Interact meeting and only one pre-IND meeting, so make them comprehensive and multidisciplinary. Third, use the correct submission channels. Meeting requests go to CBER DCC. ECTD is not required for meeting requests, though it is encouraged. ECTD is required for commercial IND sponsors. Fourth, prepare quality packages. Consider submitting Monday through Wednesday. Include clear cover letters with all required information. Limit questions to about 10 maximum, including sub-questions, and make sure pre-IND meetings are multidisciplinary. Continuing this, communicate effectively. Your RPM is your primary contact. Use secure email, include your pre-submission number, IND or BLA number in the subject line, and respond promptly to information requests. Be patient. OTP receives a lot of work, and RPMs are managing multiple complex submissions. 6, know the rules. You can cross-reference active and inactive INDs with a letter of authorization, but not withdrawn INDs. Respond to meeting comments in your future submissions, and if your research IND becomes commercial, remember to switch to ECTD format. And lastly, plan your timelines. INDs have a 30-day review. BLAs have a 12-month standard or 8-month priority review, and that includes the 60-day filing period. Meetings are scheduled within 30 to 75 days, depending on the type. And remember, commercial INDs must use E2B R3 format for safety reporting as of April 1st, 2026. Bottom line, OTP is here to support your innovation. We have the structure, the expertise, and the resources to help you bring therapies to patients efficiently. Before we wrap up, I want to make sure you have the resources and contacts you need. This slide is designed to be a reference tool. I've organized everything into web pages, guidance documents, SOPs, and contact information. And I'm not going to read through each one, but I encourage you to take a picture of the slide or visit the CBER homepage where all of these resources are linked. Key message, you don't have to navigate this alone. We have the expertise, and if you can't find what you need, just reach out to OTP or PMS. And that concludes my presentation. Thanks so much. I hope I gave you a clear understanding of how OTP is organized.
Thank you for this presentation. So let's take some questions. Are there any questions from the room? We can start with online questions.
The first online question is, an existing program has RPM. For our next new program, do we contact the same RPM or do we contact the OTP RPMS email?
That's a good question. So usually we assign RPMs depending on their current workload. So most likely you'll get a new RPM assigned. We do send the acknowledgement letter within 2 weeks of an original IND. So you should have your RPM contact within a couple of weeks. But if you're unsure, definitely contact OTP RPMS.
Next online question. For N=1 study, can we have interact meeting?
That's a good question. You could?
So I guess it depends on context. So you can send an email to OTP RPMS to ask this question within the context of your application.
Next online question. Is the letter of authorization needed to cross-reference another IND under the same sponsor?
Yeah, that's a great question. The answer is you do not need a letter of authorization, but just include that in your cover letter that you are cross-referencing your previous IND submission. So as long as it's active or inactive, then it should be okay.
Next question. How do we know if a cross-referenced IND is inactive or withdrawn?
Yeah, that's a great question too. You should be able to contact that IND holder or master file holder, and they will let you know because they do need to provide you a letter of authorization to cross-reference.
Next online question. Can a sponsor request more than one end of phase 2 or pre-BLA meeting?
No. So if it's the same product and indication, we will only grant one end of phase meeting, whether that's end of phase 1. We'll grant an end of phase 1 or an end of phase 2. You can have both of those or a pre-BLA meeting. But you can't have more than one if it's the same product and indication.
Next online question. When are sponsors notified of changes in RPM assignments for a program with a PTS number or IND number?
Yeah, that's a good question. We do sometimes have some turnover, and the best way to find out if you have a question directly to your RPM and they're not responding by acknowledging your email within 48 hours is just to send an email to OTP RPMS, and we'll help you figure out if that RPM was changed.
And also that was in one of your slides too, right?
Yeah.
So if you have any urgent inquiries,
yes.
They can send an email with a title, urgent.
Yes.
And send it to that email address.
Yeah.
Next online question is, what if I have questions after my Interact meeting, but I'm not quite ready for a pre-IND meeting?
Yeah, that's great. I would contact your RPM first with the questions that you might have. You do have available availability to ask for a type C meeting or a type D meeting, but we would encourage you to talk to your RPM first to make sure that's the best pathway. Also, if you have a clarification question, you can submit that to your PTS number or IND number, request clarification on whatever you received.
Next online question is, what if I cannot get a letter of authorization for my single patient IND?
Yeah, if you can't get an LOA, then you will need to submit full CMC and non-clinical information to your single patient IND. But if you have any questions on that, please contact OTP RPMS.
And this is all for online questions.
Thank you so much. So we really appreciate your time, and so we can continue to the next presentation. Thank you. So our next presenter is Dr. Kimberly Schultz. Dr. Schultz served as the Director of Division of Gene Therapy 2 within Office of Gene Therapy CMC, part of the Office of Therapeutic Products at CBER. In this role, she oversees product review of pre-IND, IND, and BLA submissions for gene therapy products. She also contributes stakeholder outreach, regulatory guidance documents, and serves on working group related to advanced manufacturing initiatives, both within CBER and across FDA. And please join me welcoming Dr. Kimberly Schultz.
Thanks for that introduction and for the opportunity to share with you CBER's experience over the last couple of years as we've been implementing the Advanced Manufacturing Technologies Designation Program. Over the next 30 minutes or so, I'm going to give you a crash course in the Advanced Manufacturing Technologies program, specifically explaining what an advanced manufacturing technology is for the purposes of this designation, why it is that you would be interested in getting this designation or working with a technology that is designated, and then once you understand how great it is, how you would apply for this designation. So the Advanced Manufacturing Technologies Designation was established through FDORA. So that was a congressional act in 2022 in order to encourage the adoption of advanced manufacturing technologies into drug product manufacturing. And so Congress really wanted to support this as there's many potential benefits to including advanced manufacturing technologies in drug product manufacturing, including the possibility of greater assurance of product quality and shortening the drug product development timeline. Also, there's some potential regulatory benefits with using an advanced manufacturing technology, both for industry and for us on the FDA side, in that it may lead to more efficient regulatory or the ability to more efficiently meet regulatory requirements and strengthen the predictability of regulatory expectations. One additional thing that really the AMT designation program does is provides another venue for interaction with the FDA for technology holders. And so I just want to take a second before I move on to just explain a couple different terms that I'm going to be using throughout today's talk. So when I'm talking about a technology developer, an AMT holder, developer, any of those terms, that's the entity that's actually developing the manufacturing methods that are going to be designated as an AMT. And that may or may not be separate from a sponsor or applicant. And so a sponsor and applicant is somebody who's developing a drug product for commercialization. And so that's a distinction that sometimes is a little bit confusing when we're talking through this, and I'll try and make that distinction as I go through. And so as Congress thought about how to encourage adoption of advanced manufacturing technology, they gave us some guardrails for which to implement this program in. And so they give a definition for an advanced manufacturing technology, whereas it's a method of manufacturing that incorporates a novel technology that should substantially improve the manufacturing process while maintaining equivalent or superior drug product quality. And some examples of how it may substantially improve the manufacturing process were also given, including reducing the development timeline for the drug or increasing or maintaining the supply. So this is a pretty wide definition if you really think about what was laid forth by Congress. And one thing that I really would like to point out is that the FDA includes many things when we are talking about a method of manufacturing. And so I have a footnote at the bottom of the slide, but I really want to point out that this includes analytical methods as well as just the manufacturing methods themselves. And so that's an important distinction as well. The AMT designation is not for a specific technology per se. It's not for a piece of equipment or a specific reagent that you are developing, but it's actually implementing those technologies as a manufacturing method for the use of producing a drug product. Along with giving us that definition of what the criteria was for the AMT program, we were also charged with a number of deliverables in order to successfully implement this program. And so on June 8th, 2023, we conducted a workshop between the FDA and many stakeholders in which we discussed the current state of innovative manufacturing technologies, what the barriers are in order to implement these different technologies and drug manufacturing processes, and what would encourage or incentivize the adoption of these advanced manufacturing technologies into drug product manufacturing. Taking all of this into consideration, we then drafted a guidance which was finalized in December of 2024 that walks through the specifics of the AMT designation program. I'm going to highlight throughout my talk today different parts of this guidance, but really this is a comprehensive resource that would walk you through the designation request process and the expectations for both AMT holders and for the FDA in order to successfully implement an AMT. Since then, we are going to provide an annual report to Congress as an update to this program. We did submit one in October, but the numbers that I'm going to be providing today are updated from that. So before I get into the how for AMT, first I'm going to tell you why, because that's going to incentivize you listening to what's going to make an incentive or a successful request. And so really, why would you go down this path of getting a designated AMT? What incentives are there for you, both as a technology developer and then also as a product developer? Why would you want to work with a designated AMT? And so really what we heard in that workshop is that the concern with incorporating advanced manufacturing methods into product development is that it may actually lead to a longer development timeline. So this is the opposite of what we would like to see. And so there were concerns that there would be regulatory uncertainty with new manufacturing technologies. And so one of the major benefits of working with a designated AMT is that there's early FDA interactions with applicants, product developers, in order to facilitate the adoption of these AMTs into their product manufacturing and development timelines. And so what this means is that we would prioritize having early interactions with applicants and the product or the technology developers in order to make sure that regulatory issues that may arise in the future are addressed early on. And then we would continue as needed throughout that product development timeline. Another major advantage is that at the FDA, each AMT has a designated lead. And so what this means is that there is a dedicated reviewer who, in addition to their other work, keeps up on what is going on with that specific AMT. They go to all of the meetings for the different files that have incorporated this AMT and are able to provide the FDA CMC review teams with the necessary background information and updated information specific to that advanced manufacturing technology. And so when we really think about a product development communication pathway, it often includes the sponsor or the applicant having meetings with the FDA, asking questions and getting feedback. And then when it's related to a new manufacturing technology, they relay this back to the technology developer. And I know that we all played the telephone game when we were younger, and you can all understand that sometimes there's miscommunication that occurs with that type of a strategy. And so with an AMT designation, this really formalizes the ability to bring in that technology developer to your sponsor meetings and have them as a part of the meeting so that they can be part of the direct interaction with the FDA in order to hopefully have an efficient adoption of the technology into your manufacturing process. Once again, that FDA designated lead is going to work internally also to help with that transition. So now that I've told you about the benefits of this program, let me give you some tips on what leads to a successful AMTD request. So as I mentioned, the guidance goes through this in detail. There's no way that I could go through this in the next 30 minutes. So I'm just going to highlight some of the main things that we think about as we're reviewing the requests in order to help you put together a successful package. And so two key concepts are discussed throughout our review process and are really fundamental to understanding whether or not your request would be granted for an AMT designation. First of all, is your technology novel? If we have sufficient experience with it, if implementation of your technology is not going to lead to any hiccups in product development, then we are unlikely to grant it as a designated AMT. The other thing that we are looking at throughout the review process is the actual maturity of your technology. And so this is important because we are expecting technologies that are IND ready. And so they should be able to be adopted by the field at the time of designation. And depending on what your technology is, there may be particular CGMP expectations that you should address in your request so that we're sure that it is ready for implementation. Along those same lines, although your technology may still be under development, it's important for us to understand that it will be able to be implemented to provide consistency and reliable manufacturing for the INDs. So with those two key concepts in mind, I'm now going to talk through what the actual content of the request should include. And so all of this is once again explained in the guidance with much more information on each of these different bullet points. Excuse me. You should start with a brief description of the technology and the manufacturing methods of which you are proposing for an AMT designation. This is where I really recommend that you talk about those two key concepts that I just went over, both the novelty of it and the technology maturity, in order to underpin the rest of the review of your manufacturing process. Also, when we are reviewing the submission, we're thinking about the context of use for which this advanced manufacturing technology is going to be applied. And you should clearly state that at the beginning of your request. And in many cases, this should include the product class and the dosage form of your proposed context of use. You then should go into how specifically your AMT, your proposed AMT, meets the eligibility criteria. And this should be supported by developmental data, including that from a model drug. And then lastly, please include the perceived regulatory, technical, or other challenges which you hope that this designation will help you address. And this is going to be important for us as we're thinking about the future and the interactions that we will have. So for the majority of my talk, I'm going to focus on these two parts of your request, the context of use and also the developmental data. These are, for us, some of the most important parts of your request and the places where we've interacted with requesters the most throughout the review process. And so the context of use is going to be your proposed specified purpose and manner of use for the designated AMT that will be used in developing and manufacturing of a particular drug. So this is what's really unique about the AMT designation program as compared to the rest of the way that the FDA really works. So normally we're reviewing a specific drug product for a specific indication. But that's not what the AMT is. The AMT is a technology that may be applied to multiple drug manufacturing programs. And so we need to understand what that product class that you're expecting your AMT to be implemented in is. And so that's what we mean by the specific context of use, and it's not necessarily a specific drug product. So I'm going to walk through an example of a context of use, a little bit exaggerated in order to have you think about how we really implement our thought process on this. And so I understand from a technology developer, you want everybody to use what you've been working on, right? But that's not really reasonable when you really think about what you're proposing. And so in this example, you may propose a context of use where it would be used for the manufacturing of cell therapy products, including cell-based gene therapies. Now, if you think about it, that's huge. And I've put up a couple examples in here. Everything from CAR T cells, TILs, CD34 cells, so stem cells, iPSCs, islets. Those are extremely different cell types that have very different necessities for successful manufacturing and culturing. And so in order to have a technology that would span this large group of product classes, that would be a pretty amazing technology. And if you have that, I'd love to talk to you about this after it. What actually happens is that we see that the C of U is demonstrated for a very specific product class. And so in this example, you may come forward with some information showing that your technology is able to successfully manufacture CAR T cells. So a cell-based gene therapy, T cell based. We look at the model drug information that you put forward, and we think about the different attributes that your technology is supporting. And then, as CBER often does, we think about what is scientifically reasonable with the data that you have provided to us, and we're happy to meet you in the middle when there is the possibility to do that. So in this case, if your manufacturing technology was able to support the manufacture of a genetically modified T cell based product, it's pretty reasonable that you'd also be able to use that technology to manufacture a non-modified T cell based product. And so it's a bit of a Goldilocks situation, and that's going to depend on the technology that you are putting forward. If it's an analytical method that tests one very specific attribute, then that context of use is going to be very specific for that. And so really think about that when you're putting together your package and be realistic in what your proposed context of use is. So as you heard me talk about the context of use, I was talking about a model drug. Well, what is a model drug? So for the purposes of AMTs and as defined in the guidance, a model drug is a representative drug that's used to generate manufacturing or developmental data that's submitted in the request. So what it really is what you're going to use to support your justification for the request and your context of use. And this can look different depending on what your application actually is. And so if you're developing a manufacturing method for an AAV vector, it is possible that you would develop a model drug, perhaps a representative AAV that expresses GFP. And that would allow you to generate data using your manufacturing technology in order to support this request. The other option that we often see is that as a technology developer, you are partnering with a sponsor or applicant who has a product under development in which your technology could help them. And so hand in hand with a sponsor, you are developing with their product developmental data in order to support your AMT request. You can also take a combination of these different approaches. But what we're really looking for is data to support what you are proposing. So this can include batch analysis data, process characterization data, and in some cases, a comparison with the conventional manufacturing that you're saying that you are improving with this technology that you've developed. And really, the robustness of the data and information should be aligned with the level of risk that's inherent to the process and the proposed context of use. And I can't underscore the importance of this developmental data. That really is the bulk of what your request should be, because that's going to provide us with a clear understanding of the manufacturing parameters in which you are proposing to operate, the limitations of your technology, which is important for us to understand as we're moving forward for future implementation of this technology in specific product applications. And then once again, the context of use. So with all that, you are now prepared to submit your AMTD request. The information here is provided to give you an overview of that process. And once again, it is in the guidance. So please go back and reference that for specifics. So what happens? You sent it into the FDA. We're always described as a big black box, okay? So this is what happens to your AMTD request once it is received. We will acknowledge it and review or assign a review team, similar to how Nadia just described that for our other submissions. Then we start a 180-day review clock, 6 months, kind of long, right? And so what happens is first we do a preliminary review. It's similar to a filing review. And we're looking to see if the request is complete. And this is where we do that novelty check. So going back to those key concepts that I talked about, having that right up front in your request is going to help us, because that is one of the first things that we're looking at when we're reviewing this. So great, we've checked off both those boxes. You've given us developmental data. It is determined as to be novel. We don't have sufficient inspectional experience with it. And now we can move on to the substantive review. And so this is the bulk of that 180 days. If it takes the full time, it depends on the amount of information requests that we are going to have to send in order to get clarity or additional information to understand what's going on with your actual technology and how that relates to your proposal. This is also where we are going to negotiate that context of use with you if you've proposed a broad one and it's not necessarily reflective of the model drug data that you've provided to us. And then we will issue a final determination letter. Congratulations, you got there. You're not done yet. So as I talked about in the beginning, the point of the AMT designation from Congress was to encourage the adoption of advanced manufacturing methods into product development. So really, this AMTD, the AMT designation is the beginning of your journey, working hand in hand with sponsors and applicants in order to improve product quality and supply within the U.S. And so, as I mentioned earlier, this brings us back to those meetings that are now prioritized with your AMT designation. So using PDUFA meetings in the context of those product-specific INDs or BLAs in order to facilitate the implementation of your technology in that product manufacturing is what you're going to be focusing on. At CBER, we really recommend that you submit a master file. There is a guidance that is available in order to show, go through the steps of how to do this and the different types of master files. But the importance for us is that this is going to provide now a centralized location of all the information that's going to support those cross-referencing INDs. And it's really going to streamline the submissions for you, the technology developer, when you have multiple INDs that are cross-referencing your information. One hot tip is when you are submitting a master file and it is to support CBER products, please submit it to CBER because we're the ones that need to be able to have access to that information. If you have questions about it, I recommend that you email OTP RPMS, just like Nadia had just talked about in the last talk. Okay, so now I'm going to switch a little bit and give you an idea of some of the experience that we've had in CBER over the last couple of years. So we do designated AMTDs in CBER, and these really are across a wide variety of technologies. Manufacturing methods for a multitude of different products, reagents that have been implemented into product manufacturing pipelines. And I also want to point out that last one, analytical methods. I really think that that's a place where technology could be advanced, particularly in cell and therapies. So on that list, on the last slide, you saw that we had granted ones. We also have some that we've denied. And so I think some of the best education moving forward is understanding why those denials actually occurred. So there were some instances where we denied because they were not novel or not innovative. And what this means is that either we have substantial experience with it in the field, and it's already widely known and used in the field. So in those cases, you don't need a designation in order to have your technology adapted to the field. You're already well used. There is not regulatory challenges. You've already made it. The other two major reasons why we have had denials is either that there was unsubstantiated claims of manufacturing process or drug supply improvement, either there was inadequate model drug or no model drug data, or the data was inconclusive. And then lastly, going back to the technology maturity part, we have had some designation requests for technologies that aren't yet ready to be implemented into an IND. And so therefore they aren't quite ready for the designation itself. And so we did provide some helpful information to these requesters, and we do hope that they come back with a request when they're ready. But that begs the question, when is the right time to request an AMT? And really, that's a larger question, right? As a technology developer, how can I interact with the FDA? So at CBER, we do have a couple different ways that you can interact with us throughout your technology development timeline. So really early on, when you're working through that technology design and you're not ready to implement it actually into a manufacturing process, you can come in and request a meeting with the CBER Advanced Technology team. That's your time to highlight your technology and then try and ask about things that you may need to include in your technology development from a regulatory standpoint, right? When you're developing a technology, you're often thinking about the engineering of it and the logistics of it, but there are specific regulatory considerations that you may need to take into consideration, such as if it's closed manufacturing, how are you showing that it's actually closed? If you have the correct electronic documentation in order to meet CGMP requirements so that it can be implemented into an IND and eventually a BLA. If you address these questions early on, then you will have a much more efficient technology development pathway. And so after you come talk with us, you go through method development, maybe you're partnering with a sponsor, and now you're going to have that model drug information, and you don't have to have a completely nailed down process, but you should be close, and you should have that data that you're meeting the minimal CGMP requirements in order to support a clinical trial. And that's when I would recommend that you come in for an AMT designation request. And then as you're moving through, you've been adopted by many different products, you're well established in the field. We may graduate you from the designation program. Many people see this as, oh no, I can't come in for all these meetings anymore, but it really should be a congratulations, you made it, you're big time. And this may be the time that you have validated processes and you can think about a platform designation request. And so I really recommend, once again, that you go back to the guidance. There is a Q&A section at the end, and one of the questions that it addresses is the platform designation versus an AMT designation. I do have a table here summarizing it, but really what I would like to highlight is that both of these programs were put forth in Fedora, and both of these programs have the same end in mind, right? Their aim is to increase the efficiency of drug development and manufacturing by adoption of advanced manufacturing technologies. But the time that you implement or use these different designations is different, and it's dependent on the maturity of your technology, with the AMT designation coming on early on, and then the platform designation coming in when you have a validated process. And there's some other differences that are on this table. Particularly, I'd like to point out who the requester is. Early on, it's the technology developer, but later on, you're really relying on the validation data that's in the marketing application. So it's likely the license holder. So I know that I went quickly. It was a crash course on what's been going on, but I hope that you as a field can learn from the lessons that we've learned over the last couple of years as we've been implementing this program. And specifically for a successful request, we really recommend that you focus on the specific criteria that is delineated in the guidance. And you as a technology developer need to determine the amount of information that's necessary in order to demonstrate the novelty and the maturity of the technology that you're putting forward. And I would also like to point out that it's really important that as you're thinking through that, you're honest about what challenges you think you are going to encounter as the technology is being adopted so that we are able to help facilitate those interactions successfully. I think it goes without saying that our review is based on the information that's on the request. It's not what's online. It's not what other people are telling us. So please make sure that you provide us with a clear and very well supported piece of information in order to review your request, your designation request. And then lastly, as I went through, the context of use is often refined throughout our review process. And so it's helpful for you to provide clear connections between your proposed context of use and the developmental data that is provided in your request. And so with that, I have provided my contact information and many of the same pieces of information that were provided in the last talk. But I am just one person standing here who was part of a large working group across CBER, who's been working to implement this program in coordination with our colleagues in CEDAR. And so with that, thank you for your time, and I'm happy to take any questions.
Thank you for this presentation. Any questions from the room? Any questions online? Yeah, it seems like we have a couple.
First online question is, can you please distinguish product developer from technology developer?
So the distinguishing of a product developer. So when I'm talking about a product developer, that is somebody who is manufacturing a drug. They are bringing the drug forward through the development pathway, IND, BLA. They are going to be the license holder of the commercialized product. The technology developer may be a different entity. And so that's somebody who is developing a technology in order to support that field. And so they may have a new reagent that is going to revolutionize the manufacturing of CD34 cells. They may not themselves be making CD34 cells. And so that's the difference between the technology developer. They are developing a technology to support the field, whereas the product developer is developing a specific product.
Next online question is there a path that allows for CDMO to gain AMT designations for manufacturing process or analytical methods that can be used for multiple clients?
So this is a question that we're getting a lot, is how can a CDMO who's not the specific product developer move forward to get these designations? And so I do think that the AMT designation is a possibility for them, dependent on the information that they have and how novel their process is.
Can AMT package be cross-referenced in BLA?
So for BLAs to be able to cross-reference something, it has to be supportive. It cannot be the specific information for a drug substance intermediate, or drug product. And so it really depends on what the technology is. At this time, for an IND, you would be able to cross-reference the information throughout product development. But then that would be an important question to talk to the FDA about when you're transitioning to your marketing application, about what information would need to be provided specifically in the BLA and what would be able to be cross-referenced. And so we do have situations where you are able to cross-reference information about specific reagents that are used, for instance, in a BLA, because that is not specific to the drug product or drug substance.
How often AMT holders can interact with FDA?
So this is a good question, understanding those interactions that are going to occur once you have a designated AMT. And so I really recommend, first and foremost, I'm going to reiterate it, that you submit a master file, because that is going to give the FDA the background information that we need in order to support interactions. And it also allows for you to submit amendments to your master file that may have specific questions that are important. The purpose of the AMT designation is to facilitate adoption of the technology into product development. It is not to necessarily help with the advancement of that technology or the development of the technology itself. So in general, those interactions for the AMT designation holder are going to be in the context of an application-specific meeting, some of the different PDUFA meetings that are available. I highly recommend Type D meetings for these types of interactions.
Next question is, can technology developer ask for informal meeting minutes?
I guess I don't understand the context of the question for informal meeting minutes. And so if there is a meeting with the FDA, those formal meeting minutes would be given directly to the applicant for which the meeting is specific for. And then the applicant is able to share those meeting minutes with whoever they desire.
Next online question is, can you comment on harmonization with other regions so that these new technologies can be adopted globally?
So this is an interesting question about global harmonization. And so I do know that different regulatory regions are interested in supporting the incorporation of different advanced manufacturing technologies. And I think everybody is taking a little bit of a different tilt on how to be able to encourage it in their own regulatory regions. In the case of novel manufacturing technologies, when we're looking at what is novel or what to designate, we're looking at the information that we have within the U.S. regulatory framework. But you are able to support those designation requests with information that may have been generated under other regulatory regions.
This can be the last question from online audience. If the technology can manufacture multiple products, is it better to submit multiple drug product data, even if the steps of manufacturing are the same?
Oh, this is a really good question. So this goes into the types of developmental data that you would have and how that's going to relate to your context of use, right? And so there's a couple different approaches I think that you can take for this, but you have to remember that your context of use is going to be based on the data that you provide. And so depending on how different the products are or how wide of a context of use you're proposing, that would be something that I would recommend you would look at the data you have in order to figure it out. But you may see a strategy where you have your main data with one product class, and then you have some supporting data with some additional product classes. But that would be dependent on how similar or different those product classes are. The other thing that I wanted to say is that your initial context of use can be expanded in the future by additional information provided after the first designation. And so it is possible to have an iterative process with us. You would not just be held to that specific context of use from the initial request.
Thank you so much. This will be the, This is the end of our session. Now I would like to invite our host to close this morning's session.
So thanks for sticking through actually a very engaging morning, and we had some great talks. So we will be taking a lunch break and be back here, or those of you online, be back to your computer at 1:10. We'll see you at 1:10. Thanks so much. I was going to sit you on me. But you were doing such a great job. Okay. Okay, I am given the thumbs up for getting started on our afternoon session for day 2. Welcome back, and welcome back to those of you who are online through Adobe. So exciting afternoon set up for you, and I get to introduce the man that needs no introduction, Dr. Graham Price, as our next moderator.
Thank you, Peter. So my name is Graham Price. I'm the Chief of Gene Therapy Branch 5 in the Office of Gene Therapy within the Super Office of Therapeutic Products. It's my pleasure to introduce our 2 speakers this afternoon, starting with Dr. Yongwook Choi. Dr. Choi joined the FDA in 2022 and serves as a bioinformatics reviewer in the Office of Cellular Therapy and Human Tissues within OTP in CBER. In this role, he reviews next-generation sequencing, bioinformatics analysis, and artificial intelligence machine learning applications in cell and gene therapy product submissions. Dr. Choi serves on multiple working groups focused on developing guidance for these emerging technologies. Prior to joining FDA, he was a bioinformatics scientist at the J. Craig Venter Institute in Rockville and a senior computational scientist at the Translational Genomics Research Institute in Arizona. He earned his PhD in computer science from Purdue University and holds undergraduate and master's degrees in electrical engineering from Seoul National University in South Korea. In 2025, Dr. Choi also earned a Master of Science in Artificial Intelligence from the University of Texas at Austin. And his talk today will be on the bioinformatics expectations for off-target assessment in human genome editing products. Go ahead, Yongwook.
Thank you for your kind introduction. So today I'll be talking about bioinformatics expectations for off-target assessment in human genome editing products. And by the end of this talk, I hope you will have a clear sense of what FDA expect to see in terms of bioinformatics methods, analysis. This is a quick overview. So I'll first briefly introduce bioinformatics and artificial intelligence staff, the group I work in. And then I'll give a brief look at the landscape of human genome editing products, because the diversity of genome editing products provides a direct implication for how bioinformatics strategies should be designed. And then I'll highlight some key genome editing safety concerns. And lastly, the bulk of the talk will be focused on bioinformatics expectations for off-target assessment. So the bioinformatics and AI staff sits within the Office of Cellular Therapy and Human Tissues under Office of Therapeutic Products, OTP, in CBER. And we reviews bioinformatics and AI-related information across diverse product types. And for example, for individualized neoantigen cancer therapies, we review computational pipelines used to identify patient-specific tumor neoantigens. For genome editing products, which is the focus of today's talk, we review off-target assessment, bioinformatics methods, and analysis results. For xenotransplantation products, we review genomic data related to the source animal genome and any edits performed in those animals. And we also review AI, machine learning, used in drug manufacturing. So as you can see, bioinformatics and AI are relevant across different product types. But today's talk, the focus would be genome editing. So genome editing products are diverse. In terms of the genome editing technologies used and the methods used to deliver the editing components, and where the genome editing is performed. So as you can see in this figure, there are various genome editing technologies, such as DNA nuclease, RNA nuclease, and base editors. And not shown here, but there are also prime editors, epigenome editors. And as you can see in panel B, there are various delivery methods used to deliver the editing components, like microinjection, lipid encapsulation, viral transduction, and electroporation. And another important dimension is where the editing is performed. So in vivo, directly inside the patient, or ex vivo, where the cells are edited outside the body and then administered back to the patient. So this diversity of genome editing product provides some important context, and any bioinformatics strategy and method should be appropriate to the specific editing technology and the delivery methods and the mode of administration. And I'll highlight some key genome editing safety risks. As you can see in this figure, so one of the key concerns is the potential to cause unintended DNA alteration that may have detrimental effects on cellular function. And those unintended DNA alterations can happen either on target site or off target site. And it can take various different forms, like small indels, meaning insertions and deletions, and large structural variants, such as like translocation, inversion, duplication. And we are focused on three major risks. One is off-target editing, the editing at unintended genomic locations. And this could be due to sharing the sequence similarity with the target sequence. And the second one is unintended on-target edits. So even at the on-target site, the editing outcome may not always be what was designed. So for example, if you designed an editor that changed one nucleotide base, but the outcome it may also create like small indels or even large deletions. So any unintended edit outcomes at the on-target site should also be assessed and characterized. Third one is chromosomal rearrangement. So if the editor can create double-strand breaks, then the erroneous DNA repair can join chromosomal fragment in unintended ways, resulting in chromosomal translocation or duplication, inversion, and which can have a biological consequence for the genomic integrity. So with this in mind, we will discuss the bioinformatics recommendations, and our recommendations are designed to help sponsors detect and evaluate all these risks as thoroughly and sensitively as possible. Next generation sequencing. NGS is the primary technology used for off-target assessment. And there are some key considerations for how to use it. The sequencing strategy should be appropriate for analysis goal. So for example, a small amplicon sequencing for a small region may be adequate to detect small edits, like single nucleotide change or small indels, but it may not detect larger changes, such as long insertion or deletion that may expand beyond the amplicon region. Also, the sequencing strategy should use adequate input material and sequencing depth. Usually, the off-target edit happens with much lower frequencies compared to the on-target edits. So your strategy should use sufficient input material and sufficient sequencing depth so that you can detect the low frequency events. And related to that is sensitivity. So you should provide in-house data or peer-reviewed publication to support your sequencing approach is sensitive enough to detect those low frequency variants. And also, quality control measures should be pre-specified and applied. And second area is documentation. Documentation is also important as the conducting the studies. So it should include all detail about methods and sequencing platform, including sample preparation, DNA extraction, and library preparation. And also, you should fully document the bioinformatics tools and analysis steps, and including like the tools that are used, and also the computational steps like command line interface information. And the sequencing quality metrics should also be documented and reported, such as base quality scores and sequencing depth and read alignment metrics. And all these results, especially if it's a long table, then it should be reported in a tabular format, like CSV format or Excel format, so that the data can be reviewed in a much easier way. So here's some overview about off-target analysis. I'll touch on each in more detail later. About assessment approach, it can be considered as a three-step approach. So one is off-target site nomination. So you need to identify potential off-target sites. So some sites that may have the off-target editing. And then for all those nominated sites, you need to verify. So this is called off-target site confirmation step. And lastly, for all confirmed off-target sites, you need to perform risk assessment, providing any potential biological consequence for that edit at those sites. There are some key considerations. I'll repeat this in the later slides, but first is using multiple complementary methods for off-target site nomination. And second is using relevant human cells from multiple donors. And you need to ensure adequate sensitivity of your method. And lastly, you need to evaluate the biological consequence for all confirmed off-target sites. Next is about sample. Sample is important, and you need to use multiple biological replicates to ensure the reproducibility and also capture the biological variability across samples. Human cells should be used either from patients or healthy donors. And samples should harbor the target mutation. For example, if your editor is designed to correct the disease-causing mutation, your sample should have that target mutation, should harbor that mutation, so that the on-target activity can be measured in your off-target studies. cell type is also important because the off-target risk is not determined solely by the editor's sequence recognition. It also depends on the chromosomal chromatin landscape of the cell. So the chromatin accessibility can vary across different cell types. So for the same editor, the off-target profile may be different across different cell types. So for ex vivo product, you should use the cell type identical to the cell type used in the drug product. For in vivo, the cell type should be the representative of the intended target tissue. And also, the biodistribution data should inform whether any additional off-target analysis should be performed in non-target tissues. So here I'll talk about off-target site nomination methods. As I mentioned, you should use multiple methods to nominate off-target sites. And here are 3 different categories of those methods. The in silico method, it scans the reference human genome, and with the user-provided criteria, it identifies the region where the sequence shows the homology to the target region. And it's easier to use or implement compared to experimental assays. So these are a good starting point. But the limitation is that the results may be biased by the initial assumption and also the parameters you used for running the tool. The second class is cell-based assays. So the editing is done in living cells. And since it's done, and after that, the sequencing can identify the off-target sites. And since it's performed in living cell, it can show you direct assessment, and also higher confidence. But it can be limited by the transfection efficiency or transduction efficiency and toxicity associated with oligonucleotide tags that are used in some assays. And the last one is biochemical assays. Here, the genomic DNA is extracted from cells, and then the editing is performed directly on the DNA. And sequencing can identify those genomic alterations. It has potential for high sensitivity because it doesn't depend on transfection or DNA repair pathways. But as a trade-off, it may give a high false positive hits. And as you can see, each method has its own strength and also limitations. So our recommendation is to use multiple methods to nominate potential off-target sites. And here are some key considerations when you perform off-target assessment. And as I mentioned, using multiple complementary methods for nomination. And the methods should be appropriate for editor mechanism of action. And for example, if some assay is designed to detect double-strand breaks, it cannot be used for some editors that does not create the double-strand break. And you should use the delivery method identical to your drug product. And confirmatory testing should be done for all nominated off-target sites. And you can compare the edited and unedited sample pairs and see what's the real off-target sites. And lastly, the on-target rates, edit rates in off-target studies should be comparable to the ones in your drug product. And again, you need to quantify not only the intended editing outcome, but also unintended editing outcome at the on-target sites. About reporting off-target sites. Important thing is this one. So nomination and confirmation criteria. You should clearly describe the criteria used for off-target nomination and confirmation along with your justification. And the report should contain a complete list of all nominated and confirmed off-target sites with these information. Genomic coordinates, the genomic location, and the number of mismatches, bulges against the target sequence, and PAM sequence information for CRISPR-Cas editors, and read count and the editing frequencies for each site. And the genomic annotations, whether it's in intergenic, exonic, or intronic. For intergenic sites, you should report the distance to the nearest gene and also whether it affects any regulatory elements. For exonic sites, you need to report the impact to the amino acid sequence. And for intronic site, the distance to the nearest exon should be reported, and also the potential impact for splicing. And lastly, risk assessment should be performed for all confirmed sites. Now we talked about off-target assessment based on reference human genome, but there are another consideration that is easily overlooked. So the impact of genetic variation on off-target editing. In fact, each individual genome has about 4 to 5 million variants compared to the reference genome. And those variants may make genomic sites more amenable to off-target editing. And I'll show you an example here. So let's consider the same genomic location in 2 patients. So one patient carrying a SNP, SNP, single nucleotide polymorphism, the other not. So in the patient without a SNP, it has too many mismatches to the guide RNA. So there is no off-target editing activity in this region. However, if you look at the genome in the patient with a SNP, then the number of mismatch is reduced, and this may create a new off-target activity, new off-target sites. And similarly, a variant may also make a new PAM sequence, so it may create a new off-target site as well. So we need to assess this impact of genetic variation on the off-target activity. And these are the recommendations. So regarding database selection and variant criteria, one or more publicly available genomic database should be used to collect the variants. And all types of common and rare variants should be used. And if you need to use any variant allele frequency threshold, then you need to justify why that may not affect the off-target impact. And in terms of analysis approach, the primary method would be in silico identification for those potential variant contributed off-target sites. And you may also perform in vitro assays to assess editing potential for those potential variant contributed off-target sites. About reporting, all the in silico search parameters should be reported, and also the results should be reported in a annotated list of all off-target sites in tabular format. The last topic is, major topic is chromosomal integrity. If the editor can create double-strand breaks, as I mentioned earlier, erroneous DNA repair can join two different chromosomal fragments in unintended way, creating chromosomal translocations or inversions and duplications, which may have biological consequence for genomic integrity. So these assessments should be performed, especially if your editor can create double-strand breaks. And our recommendation is to use sensitive, quantitative, NGS-based methods to detect low-frequency translocations. And it should be able to detect the translocations between on-target sites, especially when multiple on-target sites are being edited simultaneously. And also, if there's any confirmed off-target, then the method should also be able to detect translocations between the on-target site and those confirmed off-target sites. Reporting is similar, so need to provide all annotated list of chromosomal rearrangements in tabular format and the frequency of those rearrangements, and also risk assessment for confirmed off-target rearrangement. Now, how to report, how to submit the information, and when to submit the information. So different submission types expect different level of details. So, but the overarching expectation is that the off-target, the recommended off-target sites assessment and chromosomal integrity assessment should be completed before submitting the IND. And at the Interact meeting, you need to provide brief information about editor being used, and also brief description of your study strategy. And at the pre-IND meeting, we need a little more detailed information. So more detailed description of the editor and any modifications to it, and also more detailed information about sequencing strategy, including all those methods that will be used for off-target nomination, confirmation, and related sequencing strategies. And also, if some parts of the study have already completed, then some preliminary results should be provided. And at the time of IND submission, a complete study report should be provided. Two additional points. If there's a manufacturing change that affect the editing activity or the on-target editing rates, then those changes may require additional off-target analysis. The raw sequencing data and bioinformatics analysis scripts may be requested during these clinical studies or at the time of BLA submission. So here are some key takeaways. Regarding bioinformatics assessment strategy, tiered multi-method approach. So you need to use multiple methods for nominating off-target sites, and then those potential off-target sites should be verified using your confirmation method. And you should use appropriate cell types from multiple donors. And the methods should provide adequate sensitivity. About reporting, annotated lists should be provided with functional consequence of all the confirmed off-target sites. And all those studies should be completed before the IND. And early engagement is important through Interact or pre-IND to discuss your off-target study strategy. And lastly, a brief note on clinical development program considerations. So bioinformatics analysis does not end at the IND. it can inform the safety monitoring, especially for genotoxicity and oncogenic risk. And also, the long-term follow-up should be needed for all patients receiving genome editing products, because the long-term effects of on-target editing and off-target editing is unknown. So here are two guidances. So first one was published in January 2024. It has a general recommendation for CMC, non-clinical, and clinical. And the second one was published as a draft last month. It has most of the information that I discussed today, NGS-based methods for off-target assessment and chromosomal integrity. And actually, this is open for public comment. So comment period ends on July 14th. So this is the docket number. So we welcome your any comments and feedback, and so that we can refine our recommendations. And this is my contact information. If you have any questions after today, then you can reach out to me. Thank you.
Thanks, Yongwook. That was a great presentation. We'll open the topic to questions now. If anyone in the room has questions, please come up to a microphone. And while you're doing so, I'm going to take moderator's privilege and ask the first question. So how does the next generation sequencing analysis impact product control through setting specifications?
Can you repeat this?
So how does now you've done the next generation sequencing analysis to identify your off-target edits? How does that affect product control through setting specifications?
Okay. So I'll provide some examples. For example, like ex vivo editing products, for example. So you identified off-target sites. So you confirmed those off-target sites. And you have like certain percentage of editing. And now those information can be used when for the product characterization or even in the release testing.
Okay, we'll take an online question now.
Yeah, we can start with, can you discuss sensitivity of next generation sequencing and how it is determined?
I think the question was discuss the sensitivity of the NGS method. Yeah. So for example, just thinking about simple scenario, like you want to do some amplicon sequencing for certain location, which was nominated by some method, and now you want to confirm. So I think it will highly depend on the sequencing depth. So if you are, for example, sequence like 100x depth, then it may not detect low frequency events. So as you increase the sequencing depth and sequencing quality, you can have much higher sensitivity to detect low editing events. So I hope that may help.
Another online question, please.
An online question. You mentioned using multiple complementary methods for off-target site nomination. In practice, if a sponsor has already used a well-established biochemical assay and an in silico method, would FDA consider that sufficient, or is a cell-based assay always expected?
I think that's good practical questions. So short answer is it depends. So it depends on the product and also depends on the methods you already use. So the key idea is that we want to ensure that the combination of those nomination methods reduce the chance of missing some real off targets. So, and you always need to provide justification why the combination of the method you use can sufficiently capture all those potential off-target sites. So the two methods may be sufficient or maybe not. So, and you can always discuss with us through like Interact and pre-IND meetings.
Thank you. We have another online question. How does the agency distinguish between detectable off-target findings and those considered clinically relevant from a risk assessment perspective, as it's difficult to establish phenotype-genotype association?
So I think the question is about risk assessment, how you actually do the risk assessment, how you link that to the clinical consequence. So most of the time, you can use available information based on the published literature or gene expression data and genomic annotation data, and also disease mutation disease association databases. So given the confirmed off-target sites, you first need to understand whether it impacts any regulatory elements, any protein coding region, and so on. And then based on that information, you need to reach out, you need to collect all this information and provide the information to us.
Okay, we have a question in the room now.
Yes, I have a question about the on-site indel addition, and then probably if it's found out to be clinically irrelevant, so would that be considered like a significant indel addition or significant on-site modification?
So you mean on-target site?
On-target site.
You observed unintended editing outcome?
Right.
Yes, those also needs to be assessed through your risk assessment and provide like potential biological consequence of those edits at that on-target sites.
Right. Like for example, in our risk assessment, if we don't find there is any kind of a clinical relevance there, so would that be considered a significant addition? I mean, on-site modification?
So I think we will review the information you provide, but I think you need to make sure that so if there's an unintended editing at the on-target site, doesn't mean the product is not allowed to proceed. So as long as you can provide scientific justification why this is not a significant safety concern, then it can be used for clinical trial.
Okay. I think we have time for one last online question.
Yes. If IND cleared with the off-target assessment, what additional information is needed at a BLA licensure stage? And please give an example.
So off-target assessments was done at the time of IND submission, but at the BLA stage, what additional information? So most of the information should be actually provided at the time of IND submission. But during the clinical studies, if there's any safety concerns, then we may ask more other information regarding off-target assessment. And also at the BLA, we may, as I mentioned earlier, we may also ask the raw sequencing data and bioinformatics analysis script so that we may reproduce the analysis if needed.
Okay. Right. I'd like you to join me in thanking Yongwook for an excellent talk. And we'll move on with the next presentation. So now it's my pleasure to introduce Dr. Christine Harman, who is a CMC Facilities Team Lead in the Division of Manufacturing and Product Quality in the Office of Compliance and Biologics Quality. Dr. Harman received her BSc degrees in chemistry and biology at Wright State University and received her PhD in biochemistry and molecular biology from Michigan State University. In 2009, she joined CBER in the Office of Blood Research and Review, where she performed regulatory review in addition to conducting research. Her research area focused on antibody neutralization of hepatitis C virus, resulting in several publications. In 2014, Dr. Harman moved to OCBQ, where she is responsible for a review of investigational new drug and biologics license applications, as well as manufacturing supplements for a variety of biological products, including bacterial and viral vaccines, cell and gene therapies, hematologic recombinants, blood fractionation products, in vitro diagnostics, and combination products. In addition to regulatory review activities, Dr. Harman also performs inspections of facilities manufacturing biologics products, and her talk today will be titled Mitigating Cross-Contamination in Multi-Product Facilities, a Regulatory Perspective on Facility Design. Christine.
Thank you. I'm so excited to be here to talk about one of my favorite topics, multi-product facilities and cross-contamination. Most of what I'm going to talk about today is based on experience with review and performing pre-license and pre-approval inspections. I'm going to begin my story just introducing the relationship between multi-product facilities and cross-contamination and differentiating between contamination and cross-contamination. And then I'm going to focus on facility design and how it plays a role in mitigating cross-contamination by highlighting some concepts and considerations in facility design. And then I suspect the more exciting part of my talk will be a discussion on 2 case studies of cross-contamination events and how they were resolved. And then after lessons learned, I want to talk a little bit more about some manufacturing considerations in mitigating cross-contamination and contamination. And then I'll conclude with some key takeaways. So let me see So we're finding that more and more facilities are becoming multi-product facilities. And this is driven in part by the increasing demand for commercial manufacturing of cell and gene therapy products, in addition to the increasing use of contract manufacturing organizations or CMOs. And the products that are manufactured in these multi-product facilities can be from different product classes, ranging from viral vectors, plasmid DNA, bacterial expressed proteins, monoclonal antibodies, CAR-Ts, just to name a few. And what's grabbing our attention as regulators is with the increase in the number and nature of the products that are manufactured in a single facility, this increases the risk of cross-contamination. So we've become very vigilant about how manufacturing facilities are mitigating this risk. So cross-contamination is a specific concern that arises with multi-product facilities, and it refers to contamination of one product with another. And this makes it a known type of contamination, and it's distinctive from microbial contamination, so it requires specific mitigation measures. But before we talk about mitigation measures, I just want to give you a peek into the mind of a DMPQ reviewer in CBER. When we receive a submission, we're involved in the review of the facilities, and we'll look at the facility and we ask ourselves, is this a multi-product facility or a single product facility? If it ends up being multi-product, then we ask a slew of other questions and we start leafing through the submission to answer them. And this is because we need to understand the risk associated with multi-product facility. So this is our need to know. The first thing we need to know is the types of products that are manufactured in the facility, because these can indicate the type of risk that we would have to understand. The other thing we're going to want to know is the manufacturing strategy. So is it multi-suites being in use concurrently, manufacturing different products, or is it simultaneous operations in a single room, like a ballroom design, or is it campaign manufacturing with a product changeover, where it's one product at a time? The other questions we're going to ask are these open operations or closed operations, and what room classifications are these operations being performed in? And lastly, we're going to want to understand the HVAC design, which are you using a protection strategy or a containment strategy or both? And we'll talk more about that. So this is just a nice visual of looking at facility design, and that it's really multifaceted with a lot of different operational and structural elements that go into it to help mitigate risk. And we're going to talk about each of these elements throughout the talk. So the first thing you need to do when you're a multi-product facility is perform a risk assessment. And this should be comprehensive in looking at all the different structural and operational elements of your facility design. And it really is important that it be comprehensive, because this should be the basis of a cross-contamination control strategy. And this is something that you would do as you introduce a new product to your facility. So the first thing you start with a risk assessment is you understand the product type and what kinds of risks are associated with that. Is it biological origin? Does it have adventitious agents in the process, or animal materials in the raw materials? Is there an airborne contamination risk? So then this is important because it'll dictate the rest of your risk assessment when you're looking at the different other elements of your facility design in order to mitigate the risks you identify with the product. For instance, with the HVAC design, you might have to consider using dedicated air handling units versus shared air handling units. And you also might have to consider single pass air versus recirculated air, or look at different strategies for your pressure cascades to determine if you need protection or containment. Also, your procedural controls, you need to consider how your personnel, materials, waste are moving around in your facility. Are you using unidirect? Do you need to use unidirectional or bidirectional flows? Or what is the degree of overlap? And personnel gowning and movement. These all will dictate how you can mitigate risk. In addition, there's some environmental controls to consider. This would include facility cleaning and any disinfectants that you're using. Are they effective in preventing cross-contamination? And then also you consider what process controls you may need to help mitigate risk. You're looking at your manufacturing strategy versus campaign or concurrent, open or closed, and what equipment are you using? Is it single-use equipment or is it shared equipment? And if it's shared, are you dedicating it? Or, I mean, if you're using shared, or what products are you sharing it with? And if it's just dedicated to a product, all of these things need to be considered. So let's talk about the first operational structural element regarding your facility design. Let's talk about HVAC. It's a big one. So dedicated air handling units in manufacturing suites, airlocks, and corridors is optimal, but that's not always feasible in every facility. So in the cases where you're using shared air handling units, single pass air is optimal if cross-contamination is a risk. But again, this may not be feasible, but there's other options. So if you decide to use recirculated air, if cross-contamination is an issue, then maybe you would limit or restrict how you recirculate that air. Other things to consider in your HVAC design are the pressure cascades, because these can help with determining if you need a protection strategy, a containment strategy, or both. And we'll talk more about that one. And then the other thing you need to consider with your HVAC design is, are you dealing with a bioburden controlled process, or are you dealing with aseptic process? And this really just centers around what kind of design do you need in your HVAC to maintain a certain classification. So for instance, if you're dealing with aseptic processes that have open process steps, then you need to have an HVAC design that can maintain an ISO 5 with an ISO 7 background. Or if you're using an isolator or closed process with a sterile product, you're going to want to consider an ISO 8 background. So let's talk a little bit about the strategy for containment and protection. And this is mainly driven by maintaining the pressure differentials. So pressure differentials can be maintained to contain product within a room to mitigate cross-contamination, or they can be maintained to protect product from contamination from other areas. And again, pressure differentials also contribute to maintaining classifications of rooms. I just want to reiterate about the different classifications for open operations versus using isolator or closed process operations with sterile product. But for a containment strategy, This would look like the manufacturing rooms would be maintained at a negative pressure to entry and exit airlocks. And the airlocks are designed as positive pressure. So air is being pulled in the manufacturing room to contain it, where the airlock is protecting the other areas as a containment strategy. Before a protection strategy, the air would move basically in a positive pressure cascade from the manufacturing room to the corridor, and it's just protecting what's inside the manufacturing room. And then sometimes you may need to employ a containment and protection strategy. And this would be where the airlocks would be designed as a negative sink, and then you would have that would be positive pressure to the room and the corridor. So the air would be pulling from the manufacturing room and from the corridor into the airlock. Alternatively, you could have two airlocks where you have a negative pressure sink next to the manufacturing room, as in the previous strategy, but then add an additional protection with the second airlock as an air pressure bubble. So another aspect of your facility design is your material, product, and waste flows. And I'll start with material transfer from the non-classified areas to classified areas. This is really important. As you should have different stages of disinfection as materials move from the warehouse to the production area. And you should ensure that these disinfectants that are used to do these different stages of disinfection are sufficient to reduce the level of bioburden. And this is particularly important when starting from the warehouse, where you have the highest level of bioburden, to then as you get into the production area. I've seen problems with material transfer, and it usually ends up with some hits in the biosafety cabinet in the production room, and ISO 5 is not so good. So this is a really important aspect, not just for cross-contamination, but for contamination. Additionally, you want to minimize the overlap of materials and waste. It's optimal to have separate supply and exit corridors to avoid overlap of waste and material, what I call maximum unidirectional flow, but some facilities are too small or can't accommodate this. So in those cases, you would want to employ some temporal separation of materials and waste moving in and out of the production area. This can get tricky as you add additional products to your facility. As you can imagine, if you have one corridor with different production suites and you're doing concurrent manufacturing, you can have a lot of movement going on in that corridor. So it's important to proceduralize things. And then the other thing to consider with your waste flow, with your material, product, and waste flows is the waste removal from the production area. So you should think about decontaminating the waste, or if you can't decontaminate it, secondarily contain it if it's liquid waste before you remove it from the production area. It's not always optimal to have separate paths and a designated area for storage and disposal of waste. Again, if this is not optional, this is not an option, then you would want to make sure that you can try and minimize or mitigate any overlap. And this will become a little more obvious as we discuss the case studies. So I want to talk a little bit to you about personnel flows. There should be restriction of movement of personnel between areas with concurrent manufacturing. Unidirectional movement is optimal, but again, if that's not possible and you have bidirectional movement, then procedural controls can be in place to mitigate any risk of that. Additionally, gowning requirements for entry in and out of the different areas should be defined, and proper training of personnel and gowning of movement is critical as well. So another aspect, environmental control part of your facility design would be facility cleaning and the disinfectants that you're using. In general, you should evaluate and qualify your disinfectants that are used in the facility and periodically review their disinfectant efficacy. When you would do this would be if you're finding any newly found isolates, or if you're seeing a lot negative trending in your environmental monitoring. And importantly too, when you're introducing new products, you want to make sure that the disinfectants you're using are able to remove those products or inactivate those products. And with that, it's important also, if you're dealing with a product that's, say, like a viral vector, it's important to evaluate the effectiveness of those disinfectants against representative viral vectors or viruses that are manufactured in the facility. And this evaluation should be scientifically justified and documented. Now switch to another process or procedural control would be line clearance and changeover. And this is important in areas that are used on a campaign basis. And the procedures for this should be based on your risk assessment. The decontamination process is dependent on the product type from a previous campaign. So it's important to understand if your changeover procedures when you're manufacturing the product and that you're changing over, that your changeover process is effective for the previous campaign. Additionally, if you're introducing a new product, class, or platform that may be more difficult, that's important to factor into your risk assessment as well to make sure that your changeover procedure is effective as well. And then I just want to mention something about common shared areas that typically don't undergo changeover but should be considered. And these areas or general support areas are examples such as weigh and dispense areas and media and buffer preparation areas. And this is actually a good segue into talking about some of the case studies of cross-contamination. So the first one is a vector cross-contamination event. And it was basically one viral vector was contaminated with another in an upstream manufacturing stage, basically in the bioreactor. So this investigation was quite complex, and there was no definitive root cause, but there were very probable root causes that were identified. The first was they did have some overlap with their waste flow with a common area that's used for weigh and dispense of raw materials and media kitting. Additionally, at that time, there was an increase in manufacturing activity, which generated a lot of waste that they were not handling appropriately. They were actually increasing the number of products that they were manufacturing. And then the last piece of this was personnel that were not following procedures for waste removal. They were not decontaminating it or secondarily containing it when removing it, and as they were moving it past a common area. So the cross-contamination event likely occurred due to improper waste handling that contaminated a shared area. In this case, their weigh and dispense area was in the warehouse, and they were also removing waste through the warehouse as well. So some of the corrective actions that they implemented, one was they followed their procedures and made sure they followed their procedures, but this time they were not removing it uncontaminated. So they were contaminating the waste or decontaminating the waste by autoclave or chemical decontamination before removal. They reduced the overlap of the waste and material flows. So the waste was no longer going through the warehouse, but a separate path out to a separate storage area. And then they ended the kitting media. The kitting media was moved from the common area in the warehouse. They moved it to a dedicated place inside of the production area. So in the case of cross-contamination event 2, in this case, it was also a contamination in the upstream operations in the vector production stage in the bioreactor, but this root cause was different. This one was due to a contaminated micropipette, and this was a multi-product use pipette that was mobile and could be moved around. And it was difficult to clean. That was one of the root causes they also indicated. Their investigation included focusing on failures of the processes and procedures that were designed to prevent contamination. And in this case, the event likely occurred during the manipulation of plasmid preparations that are used prior to setting up the bioreactor. And it was using a pipette that had been contaminated by an operator using poor aseptic technique with the previous product. So in this case, they had a lot of CAPAs, but these are the major ones that address the major root cause. So in the first CAPA, they implemented product dedicated equipment. The second is they implemented filter pipette tips. So this was a little concerning that they weren't doing that before, but now they're doing it. And then another CAPA was they evaluated their flows, the storage hold locations for buffers, non-viral versus viral areas to identify any risk points and fill gaps there. They also evaluated their disinfectant qualification program, looking at worst case conditions, worst case viruses, decreased contact time and cleaning procedures. They actually had a very robust virucidal effectiveness study, but they were looking for gaps in any gaps that they could find as far for their corrective actions. So there's some lessons learned to be here. I learned lessons from these events as well as the facilities did too. So three main lessons that I take from these case studies is the first is risk of cross-contamination is higher depending on the process stage. So in the vector stage, and with the vectors itself, it seemed to be the upstream was really the highest risk part of the process. And in this case, adeno-associated vectors and lentiviral vectors, they seem to replicate in the same conditions. I'm not sure if people knew that until that happened, but totally different type of vectors can replicate in the same conditions. So the second is the significance in minimizing the degree of overlap of waste and materials. So improper waste handling can contaminate surfaces that overlap with raw material flow and pathways. So it's important to minimize these flows, and if you can't, to find other ways to mitigate any risk of contaminating any surfaces that are in common areas. And that the third is, and the most important, is that common areas and equipment can be high-risk areas. Because these areas do not undergo any robust changeover procedures or decontamination processes, they're often overlooked. And if there's any chance that they could get contaminated, then you kind of have a problem, because most of the focus is in the manufacturing room where you expect the risk to be. But the common areas, you don't always expect that. And a fun fact from learning about all this is actually AAV can remain viable on a surface for more than 3 weeks. So vectors, so it's something to think about depending on your product. If it's a live type of product or a replicating product, that's something to think about. And the small mobile equipment can become contaminated and are difficult to disinfect and decontaminate. So something to think about in your risk assessments. So now I'm going to spend the rest of the presentation on some other manufacturing considerations that are good in mitigating risk of contamination and cross-contamination, particularly some manufacturing technologies that we're seeing with cell and gene therapy products. So cell and gene therapy products are pretty much predominantly using single-use disposable systems. And we like these single-use systems because it reduces risk of cross-contamination and contamination, and it eliminates the requirement for cleaning, which is one of the major sources of cross-contamination if you don't properly clean shared equipment. These systems are used mainly in cell and gene therapy products because they are help with closed system processing for aseptic processes. And it's closed only if you can demonstrate that it's closed. Let's see, demonstrate, I just lost my train of thought, sorry. these systems are helpful for maintaining aseptic process. So, but in order for them to be considered aseptic process, you have to demonstrate that they're closed. And there's this can be done by verifying if the components are received sterile, and also using the components in an aseptic media simulation study. So this can also be applied to fully automated systems. And in fully automated systems where there's no open manipulations, this actually reduces the risk of multi-lot processing in a single suite. With the fully automated systems, these can actually operate also in a reduced room classification as single-use disposable systems. But with the fully automated systems, there's a little bit more to worry about in that these systems are not visible. You can't really see leaks, and the ability to detect leaks is a little bit difficult because it's in a closed, kind of a closed system where you can't really tell. And it has some technologies associated with it. So we usually ask for, we like to see risk assessment and mitigation of any potential failures and integrity of the system. We also still expect to see media simulation studies. And I think that's about all I'm going to say with that. Sorry, I lost my train of thought. But I want to talk a little bit about closed systems and aseptic processing, because we've seen some confusion and some deficiencies regarding reviews and an inspection regarding closed systems and aseptic processing. One is the definition of the aseptic boundary. So process steps that occur after the final sterile filtration are considered within the aseptic boundary. And if the entire process is aseptic, the entire process is considered aseptic if there's no final filtration step. So that means all the components that go into the aseptic boundary must be sterile, and the sterilization processes of the components must be validated. So filters that are used to sterilize media components and such that go into the aseptic boundary needs to be validated. Additionally, for the aseptic process simulation, this should cover all the process steps within the aseptic boundary. And if the entire process is aseptic, then all the steps, including the worst case steps, should be simulated. And if there's a final filtration step, only the steps after the sterile filter require demonstration of APS. And lastly, I just want to talk about multi-lot manufacturing in a production room, single production room. And this is basically processing of multiple patient lots in the same production room. This looks like multiple work streams with 2 or more biosafety cabinets within the room that are utilized simultaneously in combination with closed systems, or it could be 2 or more fully automated systems processing simultaneously in the same room. So in this regard, with this type of strategy, we would expect a risk assessment to focus on any potential contamination, cross-contamination between lots, and also mix-ups between patient lots. In addition, for the qualification of this type of manufacturing, we would expect to have maximum number of work streams and personnel implemented, and your EMPQ should have the maximum number of personnel. Additionally, the aseptic process should be performed at maximum capacity under worst case conditions. So just some concluding takeaways. We started with multi-product facilities with doing a risk assessment. So perform a comprehensive risk assessment evaluating facility engineering, process, procedure, and environmental controls, and developing a cross-contamination control strategy. Make sure your HVAC design should be appropriate for the type of manufacturing operations and product being manufactured. Disinfectants that are used in your facility should be appropriately evaluated and scientifically justified for virucidal effectiveness if applicable. Use of single-use equipment, disposable equipment, and fully automated systems significantly reduces risk of cross-contamination. The aseptic boundary should be defined and ensure all components entering the boundary are sterile and the sterilization processes of the components is validated. And basically from our lessons learned, risk of cross-contamination is higher depending on the manufacturing production stage, and common areas and shared equipment can be high risk points that should be considered in the risk assessment. And I'd just like to acknowledge some colleagues that helped with this presentation. Thank you.
Thank you, Christine. That was a great presentation. Questions if there are any questions in the room, can you make your way up to a microphone?
Absolutely.
Thank you very much, Irina Chirpanova. So is there an expectation that the aseptic process simulation, as you call it, the rigor of it would be different prior to first-in-man study versus later studies?
Oh, I'm sorry.
For completely aseptic process,
right,
from the beginning to the end, is there an expectation or allowance that the rigor of aseptic process simulation prior to first-in-man will not be as high as for the later clinical batches?
Oh, are you saying commercial versus clinical? Or
well, during clinical development, right?
Oh.
So young company going into first-in-man study, right? And they're just developing their process and closing it, right? So what's the expectation there? To do it prior to the release of first clinical batch?
I believe that APS is required during clinical because it's a safety issue.
Mm-hmm.
Yes.
Mm-hmm.
Okay. Can we have a question online, please?
Absolutely. Can you hear me? If the manufacturing process is demonstrated to be entirely closed and or uses a completely closed automated system in which there are no open steps, can the manufacturing be performed in a CNC environment as opposed to ISO 8 environment?
So that I'm ready for that question, actually. So for aseptic process, it really, the classification of a room to perform operation is dependent on risk and if the product is requiring to be sterile. So the environmental controls should be in place to ensure that sterility is maintained and product quality is maintained. And for closed systems, it could allow flexibility for a lower classification. However, our perspective, ISO 8 is what we recommend. And the reason is that there's three reasons. First is CNC is not routinely monitored, so we're not really clear on what the environmental control is of a CNC area. The CNC areas for different facilities could be different microbial levels, and some are using HEPA filtration, some are not, and so there's a lot of uncertainty in the environmental control of that. But the big thing is that because there may be leaks in the closed system that you cannot detect. And in those uncertain instances, it's better to, there's less risk if you're performing those operations in an ISO 8 versus a CNC. So I'm not saying that you can't ever do a fully closed system in a CNC. I just think it would be a hard sell for us. And you would have to have very robust scientific justification and a lot of data to demonstrate that your system is fully closed.
Okay. Let's have another online question, please.
Yes, there's another question. As a multi-product facility, what information would need to be provided in the submission to support manufacturing of multiple products? Are there limitations on the type of products that can be manufactured in the same facility?
Yeah, there could be limitations. Yes, it depends. If you have So there's certain product types that, like cytotoxic agents or antibiotics, we generally don't allow that for biologic products. I would recommend, though, to come into the agency if you have want to introduce products that are considered those type of products, potent compounds, cytotoxic agents. Those are the only limitations I could think of. But if you wanted to manufacture very diverse product classes like that, you will have to have very robust facility controls for that. I don't know if I answered the question, but.
I have a question.
Okay.
So because a sponsor may not know the other products being manufactured in the same facility, how critical are master files?
Sometimes the master files won't have that kind of information. We would have to look at, sometimes the master files do. What we end up doing is if we need to understand, like an applicant will submit something and they are using a CMO to manufacture, and we need to know, what kind of products are being manufactured at the CMO. So we can't really contact the applicant because there might be some, proprietary information there. So we end up having to contact the CMO directly and ask them. That's what we end up doing. But because master files, it's a little difficult. They may not have a master file. That might be one thing too. So not every firm has master file.
Question in the room.
Yes, I have a question about, I understand that is common areas are very problematic. So I was wondering, do you implement in a gowning change before it, excuse me, before you come to a common area or use a common equipment? That's also actually mitigate the risk of cross-contamination, isn't it?
Yeah. So I'm not saying all common areas are going to get that you need to do changeover for all of your areas. In your risk assessment, you should consider any area that would support multiple products. Is there any chance that that area could get contaminated? It's really product dependent as well, because not all products are going to be easily vectors are just really special compared to the other products. And the only cross-contamination events that I'm aware of have been vector facilities. So to me, they're the highest risk of cross-contamination. But I would just look holistically at your facility and just focus on any shared equipment, shared areas that have any potential, especially when you're dealing with different products, very different products.
Even though there's like, for example, it's the same AAV, but also the gene of interest is different. So that's also actually very, very important, even though the vector is same.
Yeah, and that's even scarier. So it's harder to identify that particular type of cross-contamination.
Thank you.
We can take one more question from the online.
Yeah, maybe this one's more kind of related. In your experience, where is the most likely place for cross-contamination to occur within a facility, and what additional mitigations or actions would you recommend to reduce risk?
Okay. I would identify the areas of risk, and then I think the previous person that asked the question, I was talking about the common areas, but to mitigate risk, I think it's best to use single-dose disposable equipment. And if you are going to reuse equipment, dedicate it, product dedicate it. That's one mitigation. And just one thing I'd mentioned regarding virucidal effectiveness, if you have multiple types of viral vectors in your facility, you might want to consider, doing effectiveness studies with the disinfectants you're using just to make sure they're effective against the different vectors types. I can't think of anything else that I think I've said. I don't know.
Okay.
Ready?
All right. Thank you, Christine.
Thanks.
So I'd like to ask you to join me in appreciation for the speakers for this afternoon and keeping to time. We will reconvene after a short break at 2:45. Thank you.
Man. You did great. I mean, compared to most of our administration, where it's not just straight institute. Yeah. Like they like, oh, we do. Yeah. It's so long. Yeah. Don't worry about it. Don't be yourself. Don't see yourself. Really. So I asked you. Yeah, I guess it can. All right, also, because I mean, you're nice. But you would, I'm just letting you know that I got to grab. So I'm putting together all kinds of comments, like top type stuff. And I think that online moderator that should speak up. She's so quiet. Barely. I actually wanted to give you more. Yeah, you were barely. And the same thing this morning, unfortunately. But Lily's so shy and quiet all the time. You can bring the microphone. Yeah, it's not like this. Yeah, that's very directional. Yeah. Just that, you were being shy and quiet like me. One more.
Good afternoon. I'd like to welcome everyone back from a break for our final speaker in the CBER track today. So Dr. Holly Brevig is a quality assurance specialist in the Division of Manufacturing and Product Quality in CBER's Office of Compliance and Biologics Quality. Inspections of facilities manufacturing biological product. She is a regulatory professional with over a decade of experience spanning biologics, pharmaceuticals, and medical devices in both government and private industry. Dr. Brevig first joined the FDA in 2015 as an ORISE Regulatory Science Fellow. She has since held several positions across the agency, including roles in the Center for Devices and Radiologic Health, Center for Drug Evaluation and Research, and previous appointments within CBER. Throughout her career, she has participated in or led numerous domestic and international inspections of facilities that manufacture biological products, including monoclonal antibodies, vaccines, and gene therapies. She has also served on a working group tasked with developing a compliance manual to guide FDA staff on conducting inspections of CDER-regulated biologics. Her private sector experience includes serving as a senior biologics and medical device regulatory expert at Harman, Phelps and McNamara, LLC, where she conducted informal audits of biologic and medical device manufacturing facilities and advised clients on responding to FDA inspectional observations. She has also held roles as a regulatory intelligence manager for U.S. Pharmacopoeia and as an FDA regulatory science fellow at Avalar. Dr. Brevig earned a PhD in pharmacology from the University of Michigan in Ann Arbor and a Bachelor of Science in Biological Sciences from the University of California, Irvine. And her talk today will be the 5 W's of CBER facility inspections for biological products. Holly, take it away.
Thank you, Graham. So good afternoon. My name is Holly Brevig, and as Graham said, I'm a quality assurance specialist in the Division of Manufacturing Product Quality, or DMPQ. So I'm just going to go ahead and start talking about the 5 W's of CBER inspections. Let me see if this works. Oh, there we go. Okay. So for the overview today, I'm going to cover why we inspect, who conducts inspections, where and when they occur, and what we evaluate, which are the 7 major systems and the 3 critical elements within each of those systems, which I'll go into detail. And then I'll also share some of the most common 483 observations that CBER encounters. So our inspection authority and requirements come from the Public Health Service Act and the Federal Food, Drug, and Cosmetic Act. So under the PH Act, a biologics license cannot be issued unless we determine that both the product and the establishment comply with the standards in your BLA and the applicable regulations. And under the FD&C Act, an inspection is needed. If an inspection is needed, it's considered to be part of the review of an application. So a pre-licensed inspection supports the review of a BLA, and then a pre-approval inspection supports the review of a PAS. And it's important to note that all the FDA Form 483 observations, if any, must be resolved before approval of the submission. So who performs FDA inspections for biologics? I'm not including CDER on here, so just CBER and OII. So CBER inspections are conducted by the Office of Compliance and Biologics Quality and the Division of Manufacturing Product Quality, or DMPQ. We work alongside our colleagues in the various product offices, for example, the Office of Tissue and Products and the others. These are typically, well, they're pre-announced and they're very product specific because they're associated with your BLA or PAS. So while we're on inspection, we're just verifying the information in your BLA or supplement. Now, the Office of Inspections and Investigations, or OII, they conduct routine surveillance inspections that can hit upon biologics. And what they're doing is evaluating the current good manufacturing practice compliance of an entire manufacturing site covering multiple products. And these are typically unannounced, and they're not product specific, right? You're just going out for the entire facility. Sometimes they'll go out for cause inspections, including based on information that, say, CBER provides them based on information in a review or a submission. And these inspections from OII are typically unannounced.
So the types of facilities that we inspect, they're across the manufacturing chain. So we'll review drug substance manufacturing facilities, drug product manufacturing facilities. I have examples of the types of processes there on the slide. We'll also inspect release testing facilities, primary labeling and packaging facilities. And one important note that we have here on this slide is that manufacturers of novel excipients are only inspected if separately filled as sterile products. If they're incorporated during manufacturing, they're evaluated under the drug product manufacturer's quality system. So this graphic shows how inspection timing fits within the review cycle. So up here, up top is the review cycle. Down here is the inspection timeline. We want to emphasize here in the next slide that it really is important that at the time of submission, your facility is inspection ready. And this is just because with the timeline for the inspection, it must fit within the review cycle. And everything that happens after the inspection, including the inspection, must happen before the action due date. And inspections typically will occur around mid-cycle, and that's within a mutually agreeable date that we've selected with the facility. So just reiterating here, inspection readiness. We expect that facility should be inspection ready at the time of the submission. And this is really just to make sure that the inspection and all post-inspection activities can be reasonably completed within the review cycle to meet this action due date. And what I have here is just some things to think about. So, significant 483 observations require time to resolve. So that's something to think about with the timeline. And any unresolved inspectional issues can delay or prevent approval. So it's good to be ready. So as part of the inspection scheduling and preparation process, we're going to reach out to the facility to request your most up-to-date production schedule. And this will help us determine the inspection dates. And that'll come through an email communication. Now, to maximize time during the inspection, we will request that the facility prepare certain documents and information to be ready on the day that we arrive. And we'll be providing a link to our SharePoint site known as the Guest Collaboration Experience for document sharing that happens during the inspection. A teleconference will be held with the facility a week or so before the inspection, and this is to discuss inspection logistics, verify that the documents will be ready, that the SharePoint site is accessible to you, to review any changes to the production schedule, and to address any questions that you may have. So just to lay out, a general idea of the inspection. So on the first day, inspectors will present our credentials to you, which gives us authority to inspect. And then for U.S. domestic facilities, we will issue a Form 482. Following that, the facility usually provides an opening presentation where you walk through your facility, the manufacturing steps, the other products that you manufacture, that sort of thing. And then we'll conduct a facility walkthrough just to get the general layout of the facility. Now, on a daily basis, we'll review the documentation, so standard operating procedures, records, and we'll interview your subject matter experts, but we'll also observe the manufacturing operations. And then we'll discuss any of the concerns and issues that come up throughout the week so that you know what's going on and it's not a surprise on the last day. So on the last day of the inspection, we'll hold a closeout meeting and we'll issue a Form 483 observation if there are any deficiencies identified. And in addition, there may be some discussion items that didn't rise to the level of a 483 observation, and those will be just shared with management. So what do we inspect? So we inspect against the Federal Code of Regulations, 21 CFR Part 600 to 680 for the biologics. We evaluate the 210s and 211s for the CGMP. And then there's other regulations depending on your product type that we'll also evaluate.
And then the big thing is we verify what you described in your BLA or supplement, what's actually happening at your facility, and that you're meeting any conditions of the licensure of that application. So what do we inspect? So there's 7 major systems that we cover during our PLIs and PAIs. That's the quality system, facility equipment system, the production system, material system, packaging and labeling systems, laboratory control system, and donor eligibility if applicable. And within each of these 3 critical elements for each of the systems, we're going to be looking at your SOPs, your training, and your records. Now, in the upcoming slides, I'm going to go through each system and what we inspect while we're on, what we review while we're on inspection. Now, the quality system is one of the most important systems we evaluate during a CBER inspection because it provides oversight of all the other systems and assure overall cGMP compliance. We start by evaluating whether your quality unit has proper organization, authority, and independence. And then we'll look at your quality manual to help us just understand the unit's responsibilities, the quality policies, procedures, and just how oversight is structured across your operations. From there, we'll examine the key elements of your quality system, which I've listed here, including the written procedures and records and any documentation you have, really to assess whether your quality unit is effectively providing the oversight required under cGMP. Now let's discuss the facility and equipment system, which is an important system covered by DMPQ. We review your facility design because the proper layout, segregation, and flows are your first line of defense against contamination and cross-contamination. So your facility should provide adequate space for orderly manufacturing, as cramped spaces can lead to errors and contamination risks. We'll look at how you've segregated manufacturing steps to prevent cross-contamination. Ideally, we'll want to see unidirectional flows, like Christine had mentioned, where personnel, materials, and products move in one direction from lower to higher classification areas. But if you can't achieve unidirectional flows, we'll need to see robust temporal segregation and/or strong procedural controls like line clearance between campaigns, as Christine had mentioned. Now, in terms of facility construction, we're looking that the surfaces are smooth, impervious, unbroken, cleanable, and durable. This is really because any kind of rough surfaces or cracks or porous materials can harbor microorganisms. And then we'll also check that the joints are sealed and that the corners are rounded just for easy cleaning. And the whole idea is to verify that your building is in a good state of repair. So moving to the systems that maintain your controlled environment, the HVAC system is critical. And I know Christine Harman just covered the HVAC design and the requirements and qualifications. So I'm not going to be really repeating that here. Briefly, during inspection, we're just going to verify that your HVAC system's properly qualified and maintained. For critical utilities like water and gases, we'll review documentation on the qualification and your routine monitoring program. What is your routine monitoring program? What tests are you performing? And then any data associated with that. Your building monitoring systems are also evaluated. These systems should be recording critical environmental parameters, and you'll need alarms and notifications for when these parameters go out of specification. So we'll be verifying that the alarms are functioning and that the building monitoring system is properly calibrated. Now let's talk about equipment. So we'll review your major equipment qualifications, including those conducted as part of the process performance qualification or PPQ, mainly just to check that the equipment is qualified for its intended use before you use it for production. We'll also be reviewing your preventative maintenance and calibration program. So those protocols that you have should lay out what the plans are for your preventative maintenance, what activities you're going to be doing, what calibration activities you're going to be doing, and then the scheduling should say when those activities should occur. And then we'll look at logs to verify that what you plan to do actually happened. Regarding facility and equipment cleaning, we'll review your cleaning and disinfection efficacy studies, And these, like Christine was saying, should demonstrate that your agents are effective against the organisms you might encounter in your facility. We'll also review your cleaning procedures just to ensure they're detailed enough that different operators can execute them consistently. And then we'll look at the routine cleaning logs just to verify that it occurred as planned. Lastly, we'll review any line clearance and changeover procedures and any documentation verifying that those occurred. And as you know, poor line clearance can be a common source of cross-contamination. So that's what we're verifying there. So environmental monitoring is how you're going to verify that all the facility and equipment controls that we just discussed are actually working. So we'll be looking at your environmental monitoring performance qualification or your EMPQ. We'll look at the protocol and the report. A big thing is checking that the sampling locations and the types of sampling that you'll be doing were based on a risk assessment. And then we'll verify that you perform any clean room requalification after a shutdown or any kind of major modification to your clean room area. So for example, if you install new equipment or you even modify the HVAC system, or you make room changes like structural changes. Next, we'll review your environmental monitoring program, including the procedures and the sampling plans. I've highlighted here that for monitoring, this should include air sampling, non-viable, viable air, as well as surface sampling of those critical surfaces in your facility and of personnel as well. So we'll also be looking at your EM documentation, reports, and the trending that you do to ensure that you are performing all the different types of monitoring you have, and that the sampling locations are continuing to be appropriate based on any data you've collected in the past and your updated risk assessments as you trend. And then we'll also be looking at whether you're responding appropriately to any excursions you have through your deviation and your CAPA system. So for the production system, our team focuses heavily on the live observation of manufacturing operations. And this is to verify aseptic techniques and process controls and other aspects match, your procedures. So as part of this, we'll be reviewing your manufacturing process validation, including the batch records. And again, this is just to verify that, say, critical process parameters are documented, but also, if deviations happen, how are they handled and what's in the batch record and what's indicated about that. We'll also look at the manufacturing validation reports too. For the aseptic simulation program, if applicable, we'll be looking at things like the operator gowning qualification records, the personnel monitoring records, smoke studies, and also the batch records for the APS qualification, and then the relevant APS protocols. And if you have any reprocessing or reworking steps, we'll also review those procedures and any associated documentation as well. For the material system, we'll be reviewing your warehouse and inventory system and SOPs, just to verify that you're properly segregating areas for quarantine, released, and rejected materials. These need to be clearly separated to prevent mix-ups. We'll also check that the storage conditions are appropriate and monitored, and that you have good inventory records and traceability. So we'll also be looking at incoming materials and release, including your supplier qualification program, how you qualify and manage your suppliers. We'll also look at the risk assessment to determine what testing is required for incoming materials. For critical components and raw materials, we're verifying that you're performing identity testing and not just solely relying on the supplier certificate analysis, unless you've set up some kind of periodic verification of the supplier's C of A through your own testing. And then we'll also check that you have proper release procedures and documentation. In terms of shipping and distribution, we'll review your shipping validation studies to really verify that you've demonstrated your shipping containers maintain appropriate temperature control and that container enclosure integrity is maintained during transit. We'll also look at your shipping documentation to ensure that there's traceability from your facility to the end user. Now, for the laboratory control system, this is critical for ensuring your product meets the quality standards. As part of this review, the QC labs should be physically separated from the manufacturing areas with a separate entrance to prevent cross-contamination. The lab should be clean and controlled environment that's appropriate for the testing you're conducting. For example, if you're doing sterility testing, you would likely have a BSc, an ISO 5 BSc. We'll also check that your equipment qualification and calibration records are complete and accurate, and just really to ensure that your instruments are working properly and that they're going to give you the accurate results. We'll also review your analytical method validation documentation to verify that your test methods are appropriate and reliable. And a big one is we'll review how you handle out of specification results and your OOS investigations. It's really important because it shows us how you identify and address quality issues when the test results don't meet your specifications. Now, the labeling packaging system is another critical area we evaluate during inspections because labeling errors can have consequences for patient safety. So we'll start by reviewing the approved written procedures and documentation for how you handle the labels. This includes how you store the labels securely, how you print them, how you track their usage, and how you reconcile them at the end of each packaging run. So label reconciliation is important as you need to account for every label to make sure none went missing or were applied to, say, the wrong product. We'll also look at your line clearance procedures to verify that you're checking the packaging line before you start a new run, just to make sure there's no materials from the previous product left behind. And we want to see what controls you have in place to prevent label mix-ups. We'll review, again, your equipment qualification records to ensure that your labeling and packaging equipment is properly qualified and functioning as intended. And we'll also review whether your process validation for labeling and packaging operations focus on whether the temperatures are maintained within acceptable ranges and labels adhere properly under the real world and distribution conditions. So now let's talk about the most common 43 observations that we encounter. Okay, there we go. Okay, so I have examples of the most common observations across all the major systems in these slides. But in the interest of time, I'm just going to highlight a few key areas, and you can review the list in the deck later. So for the quality system observations, the one consistent observation that comes up is a lack of quality oversight. This is critical because your quality unit is your independent check on the entire manufacturing operation. When oversight fails, problems can go undetected and unaddressed. The most common problems we see are inadequate deviation management. So deviations are not opened or they're not investigated properly. So there's examples of seeing something in a batch record where something went wrong, but then there's no, documentation of why a deviation wasn't opened, kind of thing. And then we've been seeing some issues with recognizing trends and deviations too. So when you don't recognize the trends, you miss any kind of warning signs of systemic problems, and you treat the recurring issues as isolated incidents, so they don't end up getting fixed. And often CAPAs don't address all the contributing factors or root causes. And then another one is there's sometimes no plan to verify CAPA effectiveness or to really evaluate whether an effectiveness check is needed. So without effectiveness checks, you have no way of knowing whether your CAPA actually solved the problem. And the same problems will just keep coming up. Now for the facility and equipment system observations, we frequently see physical degradation of cleanroom surfaces, and this is problematic because those cracks and deteriorating surfaces create areas that can't be effectively cleaned, and then they become sources of contamination. Miscalibrations in maintenance means you're operating equipment without knowing if it's performing as intended, and your critical process parameters might be drifting out of specification. Observations related to EM and EMQ are also commonly observed, such as a lack of air monitoring or sampling sites that don't represent the sites where the most critical operations are performed. EM is really your verification that your contamination controls are actually working. So if your sampling sites don't represent where critical operations occur, your data won't tell you whether the product is manufactured under controlled conditions. Now for the production system observations, the most common issues are a failure to follow procedures and document properly in the batch records. Now when operators are deviating from procedures without documentation, you've just introduced variability into your process and you don't really know how it's going to affect product quality. Another one we have seen a lot is aseptic practice that compromises sterility assurance. For example, we've seen, someone touch the floor, an operator, but then continue working and manufacturing without changing their gloves. We also see aseptic process simulations that don't simulate all the critical aseptic processes. For example, we find facilities that did not simulate where their product was exposed to the environment. So you want to include all those critical aspects in your APS. For the material system, one of the most critical issues we see is facilities relying entirely on the supplier's certificate of analysis without ever performing their own testing. So in this case, if you're doing this, you're assuming the supplier is tested correctly, but you're doing it with no independent verification. So if they send the wrong material or you don't perform the identity testing, it's going to go into your product. So you should conduct identity testing on critical incoming materials and periodically verify the supplier C of A's through your own testing. Another common issue we see is that materials are not properly segregated. So when quarantine, released, and rejected materials aren't clearly separated, you risk using unapproved or rejected materials, and that could lead to patient harm. For the laboratory control system observations, the most serious issue we see is testing into compliance. Facilities perform repeated testing and use passing results to release product without investigating the initial OOS result. This is problematic because you're potentially releasing defective product or missing issues in your manufacturing process. We've also encountered issues with document control and data integrity. So, for example, data from a building monitoring system was deleted with no backup, or there's no controls to prevent unauthorized changes to data records. So if your GMP decisions, like your batch release investigations, are based on data you can't verify or reconstruct, you can't demonstrate the integrity of those decisions. For labeling and packaging, the most significant issue we've encountered is inadequate shipping validation. We frequently see facilities that haven't performed a shipping validation at all, or their studies don't represent worst case conditions. This means you don't know if your product can withstand the temperature extremes and physical stresses of the actual transportation. We've also seen validations, shipping validations, where the container closure integrity failed during transit. For sterile products is particularly serious, as cracks and failures can mean potentially contaminated product reaching patients. We've also seen poor control of label templates and excess printed labels. This can create mix-up risks with patient safety consequences, wrong dose, wrong product, or missing safety information. So let's talk about how to respond to 483 observations. So you should respond within 15 business days. That excludes weekends and federal holidays after you receive a 483, if you do receive a 483. Now, where to send the responses? We just updated our process, so this slide isn't up to date or reflective of the process now. So we created a email address that is specific to CBER DMBQ that you can respond to. It'll be on the Form 483 and any lead inspector will let you know about it. But at the same time, applicants, you can still have the ability to submit to ESG, to your BLA or supplement And if you're a CMO, we're hoping that you can use this email address, but there's still an option to submit with applicant or separately from the applicant to ESG, or you could use CBER's Document Control Center. So there's a draft 483 or draft guidance on how to respond to 483s. You can check that out for more detail about the best practices for responding to 483 observations. You can also put your own comments on what you think of that draft guidance. But just briefly here, the responses should be comprehensive and well organized. And you should provide adequate CAPAs that address the deficiencies with the time frame of implementation and completion. Ideally, the CAPAs should be implemented before the action due date, but we realize that some CAPAs, it would not be possible to complete in the time frame of the action due date for your file. So it's important that we just know when the CAPA will be implemented so that we can assess the risk prior to the action due date. So in terms of take-home messages, the three big ones are, okay, CBER conducts PLIs and PAIs, and your facility should be ready at the time of its submission for an inspection. We try to conduct the inspection around the mid-cycle review, and all FDA Form 43 observations, if any, must be resolved before approval of your submission. The other takeaway is just the 7 major systems that are inspected during the PLI and PAI, and the 3 critical elements, your SOPs, training, and records. And then the most common observations that we find, just in summary, are the quality system, repeated deviation, not checking CAPA effectiveness, or inadequate procedures and qualifications of the facility equipment and or validation. And I want to have a special thanks to Christine Harman, who helped, and Wei Wang for developing this, I think initially, and then I just added it and changed it, Tony Lorenzo and Melissa Mendoza. So thank you for your time.
Thank you, Holly. Do we have any questions in the room? And while we're waiting for someone to be brave and step up to a microphone, I'm going to ask a question. Are there any special considerations for DMPQ in conducting overseas inspections or other considerations that need to be taken into account for those?
Yeah, so I guess for, I think most of the inspections we've done, and other people from DMPQ can chime in that are international, we do try to ask that the documents are translated if possible in English, but we know it's not always possible. So usually there's a translator there to help us walk through any of the questions that we have while we're on site. And I think similarly, it's harder when you're saying some other countries to even have the translation. So we rely a lot on the translators, but I think that's the only thing I can really think of.
Great. We'll take a question from Rebecca from online.
Yes. Can you hear me?
Yeah.
Yeah.
Okay. So here is a really, I think, critical question, relevant. If we indicate on Form 356H that our facility won't be inspection ready at the time of submission, how does that affect the review timeline? Will FDA wait for us to be ready, or will it impact our approval date?
Yeah, that's a good question. So it is still really important to be ready at the time. If you can't be ready, it's possible we could talk about when you would be ready and whether that could fit before the mid-cycle of the submission. We have had cases where that happens, and it could turn into a CR because the facility is not inspection ready. We've also had other facilities that'll withdraw and then resubmit when they are ready.
Thanks, Holly. We'll take the question in the room.
I have a question related to the timeline. So it's like for PAs, like for the supplement, if inspection is required, could you please clarify review timelines? Is it 2 months for filing period followed by 6 months? if the inspection is required?
So I think typically for CBER, they're the PAS, so that's a 4-month cycle. So we would shoot for the month 2, the end of month 2, early month 3.
So it's 2 months to make sure filing is correct, and then within 4 months, everything is.
Oh, I see. So the filing decision for a PAS, I don't actually recall what the timeline is for that, but I think it's shorter. I think it's more like 30 days.
Yeah, sounds good.
Yeah, so the filing decision is different because it's based on what is in your submission and the information. So anything in the ECTD and whether it covers what reviewers would need to make a decision. We do have a timeline for whether we decide to go on an inspection or not before we even reach out to you. And that's also around the filing meeting decision. So that's before we would go on inspection. So usually we would have the filing decision, decide whether we're going to go on an inspection or not based on discussions we have within the team and our colleagues, say in the product office, like OTP. And then after that, then we would notify you whether we're planning on coming, and then work out a schedule of when we could come, ideally mid-cycle.
Everything is completed in that 4 months.
Yes, and it has to be completed within the 4 months. Yeah.
And one thing I'll add is that GMP inspections are not necessarily going to be required for all PAS submissions. But when they are, the timelines can actually be very challenging for us because there's a lot of moving parts that need to be organized in quite a short time.
Right. That's why I think, especially for the PAS, it's important to know that when you're submitting the file and you're indicating your facility is ready, that you have in mind that, okay, maybe around the mid-cycle is when they may come, and what do we have in production? And if not, again, we can work out a different time to come, or within, that, of course, we can get everything done in the action due date timeline or before that. But you also can have the option of, if possible, creating mock operations for us to observe.
Can I ask a follow-up? So with the challenge, do you consider remote inspection if it is, ex-US facilities?
Oh, sorry, can you repeat that?
Any remote inspection?
Oh, I don't know if we're really doing that anymore, just because of it not being really a public health emergency anymore. Yeah, that's the answer I have for you that I know of.
Okay. We'll take one more question from the back of the room.
Yeah, here is a good one. If a CDMO is used for BLA, does the sponsor get information or get informed does the sponsor get informed for a CDMO being inspected for the PLI or pre-licensure inspection?
So I think they should end up finding out. That's a good question. So, okay, so if the lead inspector goes through the regulatory project manager, and that regulatory project manager will notify the applicant of any questions we have about the inspection, usually the production schedule, and then that's kind of a highlight of, okay, now we have the production schedule and the applicant knows. So now we immediately directly email the facility or the CDMO. Sometimes we do directly email the CDMO. We're usually just assuming in those cases that the CDMO is contacting the applicant, indicating that we are coming.
Okay, I think we've got time for one last quick question.
Oh. What if your facility is inspection ready, however, your production cycle does not align with your PAS review cycle? Is it recommended to delay an important submission to ensure production alignment?
That's a really good question. I think we don't want to be too disruptive to, your production schedule and your plans. So in that case, we may go a little earlier or we may go a little later. So we do try to be flexible. It's just a goal of doing mid-cycle.
Okay. I think that's all we have time for. So I'd like you to join me in thanking Holly for an excellent talk. And I'd like you also to join me in thanking all of our speakers, moderators, and backroom and front room crew for organizing a very good series of presentations today. Thank you, Holly.
Thank you. Thanks, everybody.
And now I'll turn it back over to Peter for closing ceremonies.
Thanks. So I'm just gonna, don't want to take your, thank you. Thanks, Graham, for, I mean, he kind of preempted most of what I was going to say, not all of it, but a little bit. But thanks to the speakers for all of their hard work. And you know, I think that for quite a few of them, it was maybe their first time speaking in front of such a large group. And so thank you for all the hard work that they put into that. Thank you to the podium moderators, you know, Sharu and Zara and Graham. Graham did yeoman's work. He was the master of ceremonies yesterday, and then he came today and did a whole lot. And he was working in the back the whole time. So he's been one of our heroes here. Our online moderators today were Mark, Tim, and Lily. Thank you for Lily. We just got to get you to project a little more. That's all. No, just teasing you. And the real heroes of all of our time here over the long, you know, planning for this, putting everything together, and then sitting there and doing this. Lonnie and Stacy, thank you so much for all of your tremendous hard work on this. And from my understanding, and you can correct me if I'm wrong online, there were no glitches with Adobe Connect today. And no glitches, so they really came up and did a wonderful job. So I'm just going to jump to the next slide. Just some reminders that the recordings will be posted in the event website once available. Certificates of attendance, if you haven't signed up, make sure, hopefully they're still out there. And do your evaluation. We really, really appreciate your evaluation. Comments that you have, things that you want to see, things that you want to hear. We put this together for you, and we follow what the evaluations tell us. We didn't have one last year for circumstances beyond our control, but I know that we did take feedback from 2024 and help that in putting together the agenda for today and yesterday. So your evaluations are really critical to us. The evaluation is going to be available for 2 weeks only. So make sure to put on your calendar that June 3rd is your drop dead date. And that's all I have. We will have a period of time with the speakers here if you have additional questions for them. Otherwise, the official session will be closing. And any comments, corrections, feedback? None. All right. Thank you for attending this conference, and we appreciate all of you actually being there, and all of you that are online providing the feedback for us for building this even better next year. Thanks. Bye.